Okay, can you all hear me?
Yes, sir.
Okay. Right. Then shall we start with the discussion? So far, the highest for this paper was 29 out of 30, taken by a physical student. Okay, from the 2028 batch. Now, let's begin from the first question. The correct statement regarding the issues and statements pertaining to biology. Life on Earth formed around 3.8 billion years ago. Why is that wrong?
3.5 billion years ago.
3.5 billion years ago. Yes, thank you very much. Let this be a discussion and not just me talking overall. Okay. So it is actually 3.5 billion years ago, and that is why the first statement is wrong. Then, second one, it is assumed that there are about 1 to 10 million species in the world. Why is that incorrect?
10 to
10 to
over
100 million.
100 million, right? 10 to 100 million species are there, not just 1 to 10 million. So then this is also incorrect. Third one, consumption of natural resources causes environmental problems. Why is that wrong?
Overconsumption.
Overconsumption.
Just now, for us to survive, we need to consume natural resources. No, so consumption is not a problem. Overconsumption is the issue here. Fourth one, sustainable food production is not based on knowledge in biology.
It is based on the knowledge on the
What are the three things that, three facts that can contribute to sustainable food production?
Production of
high yielding varieties of plants and animals, disease resistant plants and animals, improving post harvest technological methods.
Very good. High yielding varieties of plants and animals, disease resistant varieties of plants and animals, and improving post harvest technological methods. So then all of them are based on knowledge in biology. So then this is also wrong. Then fifth one, heart disease is a serious health problem in which causes are not fully understood yet. So then this is a very simple question. Answer is answer number 5. Okay, right. Then let's move on to question number 2. Okay, the process of passing genes that control specific physiological, morphological, and behavioral characteristics of organism from generation to generation is known as heredity. That is the correct answer. However, what is adaptation?
Peculiarity of structure, physiology, and morphology that contributes to the
Peculiarity of structure, physiology, and behavior that ensures that
promotes the likelihood of an organism's survival
survival and
for
reproduction.
reproductive and
Yes, that is the correct one. Okay, so adaptation is that. What is irritability? Ability to respond to stimuli is irritability. Evolution is the change in the gene that happens over millions and millions of years. So that is wrong. Order and organization is how you have molecules, then organelles, and then cells, and then tissues. So then maintaining that hierarchy, that is known as order and organization. Correct answer here would be heredity. Answer number 5. So far, the first two questions are pretty straightforward and easy. Okay. Right. Question number 3. Question number 3 has no answer. Question number 3 has no answer. All of them are incorrect. That is why I have provided all. Okay. So we'll see why this question is wrong. Okay. Water acting as a thermal buffer in water bodies. Is it due to high heat of vaporization? No, it is due to high specific heat. There's a difference between high heat of vaporization and high specific heat. High heat of vaporization, use the word vapor to help you remember. Vapor means your liquid is converting it into water vapor. So that means the energy required to change water to go from a liquid to a gas, that is high heat of vaporization. High specific heat would be so much of energy is provided to change or to increase the temperature of water by 1 degree Celsius. So to be exact, it's around 4,200 joules per kilogram per Kelvin. So that is high specific heat. High heat of vaporization is the energy that you need to provide to change the phase to make water go from liquid to vapor. Okay, so acting as a thermal buffer in water bodies. So there itself, you know, it's liquid. So then this would be high specific heat, not high heat of vaporization. Second one, releasing much heat energy with a small decrease in its own temperature. So that is a property of what? That is a property of high specific heat, not high heat of vaporization. So then this is also incorrect. Then the survival of organisms in the aquatic bodies in the polar regions during winter. So survival of organisms in aquatic bodies, that would be due to expansion upon freezing, right? Such that in the polar regions, the water starts freezing. And if you take ice and compare it with water, which one has more density?
Water.
Water at 4 degrees Celsius has more density. So what happens is, in the polar regions, when the water bodies start freezing, only the top layer will freeze. Why? Because all the water which is at 4 degrees Celsius, it is much denser. So it will go to the bottom of the pond. And there will be an ice layer formed on the top. So if only the ice layer is formed at the top and the water is present at the bottom, then fish and other organisms can survive. And this ice layer at the top will act as an insulator, keeping the temperature below that ice layer at a normal level. So then this is again not due to high heat of vaporization. Fourth one, maintaining a constant temperature in living systems during the temperature fluctuations on Earth, that is also high specific heat. Last one, having a maximum density of 4 degrees Celsius, that is due to expansion upon freezing, not due to high heat of vaporization. So now tell me, what is the actual property, or what does high heat of vaporization actually contribute to?
With a minimum loss of water, it can release much heat energy. It cools the surface body of the organisms. It prevents in overheating of organisms, and evaporation of sweat from the human body maintains the constant temperature, and transpiration in plants maintains the plant body cool, and in warm times it prevents it from getting heated.
Okay, that is good. But high heat of vaporization, out of what you just mentioned, only two can be written in the exam. One is transpiration, which will cool the surface of plant bodies. The other one is evaporation of sweat from animals, which can keep the animals cool. Okay, those are the two main effects of high heat of vaporization. So then the correct answer here is unfortunately not available. That is why I have provided the answer as all. Then question number 4. Which of the following is correct regarding polysaccharides? They are large molecules but not macromolecules. Is that true?
Oh, they are macromolecules and biological polymers.
Yes, they are macromolecules and biological polymers. So then this definition is true to whom?
Lipids.
Yes, very good. Right? They are large molecules. Lipids are large molecules, but they are not considered as macromolecules or polymers. Okay. Then they all have linear structure. Is that true or false?
False.
False. All right. So then let's try to involve everyone here. So anybody who can answer my question right now, please raise your hand and I will ask that question from you. Okay. So here they say they all have linear structure. So it is false. Can anybody tell me what are the structures of polysaccharide? Raise your hands and I will choose a person to answer the question. Okay, Reema's Istikar. Can you tell me the structure of polysaccharides?
The polysaccharides can be linear and branched as well.
Okay, very good. Then we will ask another person. Amina Munsir, can you tell me what is the example of linear polysaccharides and examples of branched polysaccharides?
Cellulose for linear polysaccharides, glycogen for branched.
Okay, any more examples?
linear structure, amylase and cellulose. Branch structure,
Amylose.
glycogen, aminopeptide, and hemicellulose.
Structure is amylose, not amylase. Amylase is
amylase.
something else. Amylose. Okay. So amylose and cellulose is linear. So if you take polysaccharides, you can divide them into linear and branched. Right? If you take linear, you have amylose and cellulose. Branched, you can have amylopectin, hemicellulose, and glycogen. Right? Glycogen is given, right, as an example in your book.
Yes, sir.
So,
Right.
Yes,
So then you have linear and branched. All right. Then based on that, we can answer the third statement. Hemicellulose have a linear structure formed from the polymerization of hexoses. That is false, no, because it is not linear structure, it is a branched structure. So raise your hands to answer this question. Okay, what is the monomer of hemicellulose?
pentoses and hexoses.
Okay, it is correct. But next time I'm going to choose the person to answer. All right, so then we can involve everyone. So correct, you are right. The monomer of hemicellulose is pentose and hexoses. Pentose and hexoses. All right, very good. Then fourth one, glycogen is a structural polysaccharide in fungi, and as I have given 5 as the answer, this is obviously wrong. So raise your hands if you're willing to answer this question. What is the role glycogen plays? What is the function of glycogen? Let's go with Ammar Mumtaz. Can you tell me what is the function of glycogen? Storage in animals. Then we will go with Suha Fazil. Is there any other additional function glycogen has? Suha, is there any other additional function glycogen has?
yes, sir, in fungi.
Very good. It is not only storage in animals, it is also storage in fungi. Glycogen has a role in both animals and fungi, not just animals. Okay, but here they have mentioned it is a structural polysaccharide. So if it is mentioned glycogen is a storage polysaccharide in fungi, then this would be correct. The last one, some polysaccharides possess nitrogen, and yes, that nitrogen containing polysaccharide is chitin, and the monomer of chitin is glucosamine. And where do you find chitin? Exoskeleton of arthropods and fungal cell wall. So then the correct answer here would be answer number 5. Pretty easy question so far. Should have gotten 4 out of 4 until this point. Okay. Right. Let's go with question number 5. Select the correct statement regarding the structure of proteins. A bond that contributes to maintaining the tertiary structure of proteins is the disulfide bond formed between the side chain of amino acids. Okay, time to ask a question. Right, let's go with, okay, I'll first ask the question and then if you know, raise your hand. Can you describe the structure of a tertiary structure or tertiary level of protein? Anybody who can describe, please raise your hand. Let's go with Alina Afaf. Can you describe the tertiary structure of a protein?
Yes, sir. It's when the secondary structure coils and folds extensively to form a precise, compact, functional, unique three-dimensional shape due to the interactions between the side chains of the amino acid.
Very good. So you stay online right now. So she has mentioned that it is a precise, compact, unique, functional three-dimensional shape due to the secondary structure undergoing extensive coiling and folding, right? Interactions between side chains. To form a tertiary structure from a secondary structure, there has to be interactions between the side chains, the R groups of the amino acids. Unlike in secondary structure, there are no side chain interactions. In secondary structure, only the backbone is involved in forming the bonds, while in the tertiary structure, the side chains are also involved in forming the bonds. All right. So now, Alina, can you completely tell me what are the bonds that are involved in forming the tertiary structure?
Hydrogen bonds, disulfide bonds, van der Waals interactions, hydrophobic interactions, and I think that's all.
Ionic, no? Did you mention ionic?
No, I didn't mention
Okay, ionic interaction. So as you all saw, it is hydrogen bonds, hydrophobic interactions, van der Waals forces, disulfide bonds, and ionic interactions. These are the bonds that are involved in maintaining the tertiary structure. Okay, so let's see, a bond that contributes to maintaining the tertiary structure of proteins is the disulfide bond formed between the two side chains of amino acid. So then that is correct, no? Because disulfide bond is an correct interaction and it is formed between the side chains. That is also correct. So first one is the correct answer for the fifth question. Then second one, during protein denaturation, the primary structure is completely destroyed due to the breaking of its peptide bond. So now you have to actually focus. Whenever we talk about these interactions and coiling and folding of the proteins, except for the peptide bond, that means except for the covalent bond. Now, if you take a protein or if you take, let's say, an amino acid chain, okay, all of these are amino acids. Each amino acid are linked together by peptide bonds. Each amino acid are linked together by peptide bonds. These peptide bonds are not secondary interactions. They are not hydrogen bonds. They are not hydrophobic interactions. They are not ionic interactions. These bonds that you see here between amino acids, they are covalent bonds, and covalent bonds are strong bonds. On the other hand, when you take the bonds that are involved in forming the tertiary structure, hydrogen bonds, hydrophobic interactions, van der Waals interactions, ionic interactions, all four of them, they are weak interactions. So now you would have noticed why I didn't say disulfide, because that is still not in your syllabus, but disulfide is actually a covalent bond, but we will not think about that for now, right? So if you take these secondary interactions are the ones that can be affected by the agents that cause denaturations. The most common one that we all know would be high temperature. So these agents that can cause denaturation can only affect those weak secondary interactions that are taking place. But if you take the primary structure of proteins, it is held together by covalent bonds, peptide bonds. So then that will never break. So then second statement is incorrect. Third one, the secondary structure of proteins is formed by the formation of intramolecular hydrogen bonds between two polypeptide chains. So then it is incorrect on many accounts. So if you take secondary structure of proteins formed by intramolecular hydrogen bonds, that is all right, between two polypeptide chains. So it can't be intramolecular between two different polypeptide chains. No. So if it is between two different polypeptide chains, it needs to be intermolecular. And secondary structure itself does not have any two polypeptide chains. It is a single polypeptide chain that is found in secondary structure. Two or more polypeptide chains are found in quaternary structures. And even if you take secondary structure, so then now it is time for me to ask the question. So my question is, please raise your hand before answering. I will choose the correct person to answer. My question is, can you describe the interactions that are present in the secondary structure? Raise your hand if you can answer this question. There's a question before I ask the question. Sir, but ionic bonds are also primary interactions, aren't they? Yes, but they are not as strong as covalent bonds. Okay, so the question was describe the secondary structure. Let's go with Sahla Fazin. Can you describe the secondary structure?
The primary structure of a single polypeptide chains bends and foils as a result of intramolecular hydrogen bond between the oxygen atom of the carboxyl group and the hydrogen atom of the amino group of the same polypeptide chain backbones form the secondary structure. There are two types of secondary structures. One is alpha helical structure, example keratin. Second one is beta pleated sheet, example spider silk fiber.
Very good, right? Very good. So that is exactly how it is in the textbook also. So as she mentioned, you have to keep in mind secondary structure. It is hydrogen bonds that occur between the backbone. If it starts including the R group, then it would be tertiary structure. Okay, then this is wrong. A change in pH breaks down the intermolecular and intramolecular interactions of the myoglobin protein. Now, why is that incorrect? Fourth one. Raise your hands if you can give me a review for that. Statement number 4. Okay, Pranati, can you tell me?
Yes, sir. So the intermolecular and intramolecular means that is quaternary structure, sir. The myoglobin protein is a tertiary structure. So a change in pH in the intermolecular interactions can't affect the myoglobin protein as it is a tertiary structure.
Very good, right? So change in pH, that means extreme, low pH or high pH. Yes, they can damage inter and intramolecular interactions. However, not for myoglobin. Why? Myoglobin does not have intermolecular interactions. For you to have intermolecular interactions, you need to be a quaternary level protein, but this is a tertiary level protein. Myoglobin is a tertiary level protein. Okay, so then this is wrong. The alpha helix structure is formed due to hydrogen bonds between the R groups of amino acids. Like I mentioned, alpha helix is secondary structure. Secondary structure does not have interactions between the R groups. So then that is incorrect. Right. So then question number 6. All right. Question number 6. Select the option containing correctly matched organic compounds, their building units and functions. So first one we have hemicellulose. Building units is pentoses and hexoses. Function is to prevent desiccation. Hemicellulose, their building unit is pentose and hexoses. That is correct. However, preventing desiccation is a function of keratin, not hemicellulose. So then that is why the first statement is wrong. Second one, what is desiccation, by the way? Desiccation is extreme water loss. Second one, fats. Fats are made up of glycerol and fatty acids. Yes, that is correct. Maintaining the fluidity of the plasma membrane. Fats maintain the fluidity of the plasma membrane is incorrect. The ones that maintain the fluidity of the plasma membrane is phospholipids mainly. Cholesterol can contribute to the fluidity. Okay. So fats and glycerol and fatty acids. So that is why that is incorrect. Third one, immunoglobulins. Immunoglobulins is a protein. No? Building unit is amino acid. The building unit of proteins are amino acids. So then that is correct. And what is the function of immunoglobulins? To neutralize foreign bodies. It is part of your, okay, that is a bit too much since we are still in the beginning part. Okay, it's part of your defense system. Formation of immunoglobulins. Certain cells are there which can produce immunoglobulins, which can neutralize foreign bodies. Okay, so then the third statement is the correct one. However, let's go to the fourth and fifth. RNA, building units are ribonucleotides, correct. Transmission of information between generation and generation. So then that is wrong, because for you to transport information from generation to generation, that is actually the function of DNA. DNA is the one that goes from one parent to the offspring, and then once that offspring becomes a parent, it goes to the next offspring. So going down generation is a function of DNA, not RNA. So then DNA deoxyribonucleotides, that is also correct. Providing site for polypeptide synthesis, that is incorrect. The one that provides a site for polypeptide synthesis is actually rRNA, ribosomal RNA. So based on that, the correct answer would be answer number 3. So up to now, 6 out of 6 should have been very possible, because all 6 questions are pretty basic. Okay. Right. Then we have question number 7. Following are the steps for observing an onion epidermal peel under a compound light microscope. Select the correct sequence. While I was making the paper, I forgot to put it as A, B, C, D. Must have been a bit difficult to read. Anyways, place the slide on the stage of the microscope facing the cover slip on top. So now this is how you do a sequence question. You don't even need to know the theory. If you have common sense, you will be able to answer it. First, read the question properly. Place the slide on the stage of the microscope facing the cover slip on top. Place the slide on the stage of the microscope facing the cover slip on top. So if you remember how to use a microscope, what they mean here is, if you have the microscope and here you have the slide, which you have, what are they using here? Onion peel, okay? You have an onion epidermal peel on a slide, and that slide you're placing it on the stage. That is the A part that they're saying, with the cover slip on top. Okay. B, cover the specimen holding the cover slip at the edge of the water drop. Cover the specimen holding the cover slip at the edge of the water drop. Okay. So if you remember, now this is the theory part. When you have the slide, once you place your specimen, you're putting a water drop on it to prevent it from drying out. And then you put the cover slip, take a mounting needle and try to put the cover slip on the stage itself so that there will not be any air bubbles. Okay. Place the thin epidermal peel in water in a petri dish, that is C. Transfer the specimen into the drop of water placed on the center of the glass slide. Okay. So now, let's say that you don't know anything about microscope. How are you going to answer this question? Take a look at the options. Take a look at the options. So B, D, C. These are the three possible things that can come as the first one. Either it could be B, D, or C. So let's take a look at B. Cover the specimen holding the cover slip at the edge of the water drop. Cover the specimen holding the cover slip at the edge of the water drop. Place the slide on the stage of the microscope facing the cover slip on top. Okay. If you see B and A, C and D, you can clearly figure out which one should come first. So here itself they're talking about the microscope. Here also they're talking about the microscope. Here they're talking about the epidermal peel being in a source, a petri dish. Transfer the specimen into the drop of water placed on the center of the glass slide. So then based on this itself, you should be able to identify that C is actually the first step. Why? Because you're taking the epidermal peel from a source, a petri dish. Okay, so then based on that, you can remove the second one. So out of 3, 4, 5, how do we decide which one is the correct one? So if it is in a source or if it is in a petri dish, you need to transfer it from one location to another. No, you need to transfer it onto a slide. Only then you can see it through a microscope. So transferring should be the obvious next step. So what is the next step? Anything that is mentioned transferring? Yes, B part. Transfer the specimen into the drop of water placed on the center of the glass slide. So then D has to be the second one. So this one cannot come because it has B. Then what? The only thing that is remaining is A and B. Place the slide on the stage of the microscope facing the cover slip on top. Cover the specimen holding the cover slip at the edge of the water drop. So A means the slide is there. Cover slip is also placed, not placing, placed. B, you are placing the cover slip. So placing is present tense, placed is past tense. So if you take B, B has to occur first, right? First you have to cover the slide with the cover slip properly, and then only A can happen, where you have to place the slide and the cover slip on the stage so that you can see it clearly through a microscope. Okay, so the correct answer here would be answer number 4. All right, so this is also pretty simple. So far, not a single difficult question has come. Right. Question number 8. The cell wall of all prokaryotic cells is composed of peptidoglycan. Okay, that is wrong, it seems. Why? Raise your hands if you can answer this question. Why is the first statement wrong? Okay. Zaka Azamdeen, can you tell me why the first statement is wrong?
Present in bacteria, cyanobacteria, in polysaccharide and protein are present.
Can you speak a bit louder? We're not able to hear you.
Peptidoglycan is present in bacteria and cyanobacteria, and polysaccharide is present in polysaccharide and protein is present in archaebacteria.
Yes. So if you take peptidoglycan is present in domain bacteria, while proteins and polysaccharides are present in archaea. Okay, so then that is why the first one is wrong. Second one, subcellular components can only be seen in eukaryotic cells. That is also false. Why? Because one example of a subcellular component is ribosomes. Ribosomes are found everywhere. Ribosomes are found in all, not almost, Ribosomes are found in every living cell. Okay. So if you take prokaryotes, they have 70S ribosomes. Eukaryotes, they only have 80S ribosomes. Is that correct? Anyone?
No.
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 Why? Because eukaryotes have both 70S and 80S. 70S is found inside the chloroplast and the mitochondria. 80S is found in the cytosol bound to endoplasmic reticulum. All right. Then third one, photosynthesis can be seen in both types of prokaryotic and eukaryotic cells. Yes. If you take eukaryotic cells, it happens inside the chloroplast. If you take prokaryotic cells, it happens in internal membrane. So then this is correct. Fourth one, some eukaryotic cells have the ability to fix nitrogen. No eukaryotic cells. Zero eukaryotic cells have the ability to fix nitrogen. Fifth one, circular DNA is freely present within the nucleus of prokaryotic cells. Okay. Circular DNA. Correct, no? So why is the fifth statement wrong? Raise your hands. Sarah Farhan, can you tell me why the fifth statement wrong?
It's not freely present.
Okay, that is not the correct answer. We'll go with the next one. Savita, can you tell me why? If you are talking, we cannot hear you. Okay, can anyone tell me why fifth one is wrong?
It is not within the nucleus.
Yes, very good.
The nuclear region.
Within the nucleus. Okay, it is found in the nucleoid region. It is found in the nucleoid region. That's why the fifth statement is incorrect. Okay. Select the correct statement regarding the plasma membrane. Consists mainly of carbohydrates, phospholipids, and proteins. Why is that wrong? Raise your hands. Suha Fazil, can you tell me why is that wrong? Hello.
It doesn't contain carbohydrate.
It contains carbohydrates, okay, but mainly of phospholipids and proteins. There are carbohydrates present in the plasma membrane. Okay, my next question is, what are the carbohydrates present in the plasma membrane? Raise your hands. I'm going to choose a person. Midda Muhammad, can you tell me? Ah, no, she put her hand down. Then we'll go with Naziha. Can you tell me what are the carbohydrates present?
Glycoproteins and glycolipids.
Yes, glycoproteins and glycolipids. Glycoprotein and glycolipids are the carbohydrates present in the plasma membrane. So let's take a look at this structure of plasma membrane. So you can see here phospholipids. So then they are the main components. Here phospholipids are present. Phospholipids are present. Okay. Then you can see proteins are present. Here peripheral protein, integral protein, transmembrane protein, peripheral protein, integral protein or transmembrane protein. So proteins are present. What about the carbohydrates? Take a look. Glycolipid. Once it is originating from a phospholipid, it is a glycolipid. If it is originating from a protein, it is known as a glycoprotein. Glyco is the carbohydrate chain. Okay, branching carbohydrate chain originating either from the phospholipid or protein. So here carbohydrates are indeed present in plasma membrane. However, the most common one would be the proteins and the phospholipids. Okay. Transmembrane protein act as pores that allow ions and some non-polar molecules to pass through. Non-polar molecules or hydrophobic molecules, they don't need a transmembrane channel to pass. Hydrophobic molecules can directly cross the plasma membrane. So if plasma membrane is a phospholipid bilayer, it can directly cross the phospholipid bilayer. So if you take carbon dioxide, CO2, okay, CO2. This is the structure of CO2, right? It's a linear structure, so then the dipole moments cancel out, and therefore it is a non-polar molecule. non-polar molecule. Carbon dioxide can easily cross the plasma membrane, doesn't even require a transporter like a transmembrane protein or a channel that is present in a transmembrane protein. So transmembrane protein act as pores that allows ions, yes, and polar molecules to pass through, not non-polar molecules. So that is why the second statement is incorrect. The composition and function of two sides of the membrane are the same. We just saw glycolipid and glycoprotein was found on the extracellular side. So then obviously the composition is different. And if glycolipid and glycoproteins are found on the extracellular side and they have their own function. So therefore both the composition and the function of the two sides of the plasma membrane are different. So then this is incorrect. Now, in this question, I have taken two answers as the correct answer, but the most correct answer would be answer number 4. Since selectively permeable, it regulates the exchange of substances necessary for survival. If you take plasma membrane, it is impermeable for, let's say, polar and ionic molecules. Therefore, it needs a transmembrane protein or a transporter or a channel to transport these substances. It is permeable to non-polar components. Okay, so you can see it doesn't just let anyone come in. Only the necessary things can come inside the plasma membrane. So that is why this selectively permeable is an important trait. Then, fifth one, cholesterol molecules provide the fluid nature to the membrane. That is correct. Cholesterol molecules does provide a fluid nature to the membrane. Now, my question to you is, let's see whether anyone can answer this. Cholesterol molecules provide the fluid nature to membrane of all eukaryotes. Okay. Raise your hand if you can give a review to this statement. Okay. Let's go with Amna Azam. Okay, Amna Azam, can you tell me why?
It occurs only in the animal cell membranes.
Very, very good. Cholesterol molecules are present only in animal cell membrane. So now if I had a question, let's say in a structured essay paper, you are asked to draw the plasma membrane in the given area, but instead of just saying plasma membrane, they told draw the plasma membrane of a plant cell. If you go and draw cholesterol, they will mark it wrong and may give negative marks. Who knows? Right? Because cholesterol is found in animal cells and not in plant cells. Very good. All right. So then, based on that, we have covered question number 9. Question number 9, 4 or 5, both can be taken. But since cholesterol molecules provide the fluid nature to the membrane only in animal cells, this is okay, but the best answer would be answer number 4. If I was doing the exam, I would know that both 4 and 5 are the correct answer, but 4 is the absolute best answer. So, answer number 4. Then question number 10. Which of the following is correct about the central vacuole found in plant cell? Stores water and other materials such as sugars, ions, and pigments. So then that is direct from the textbook. If you take the central vacuole, ions. Can you tell me what ions? Raise your hand if you can tell me what ions are present. Okay. Fazila Fawaz, can you tell me what ions are present in the central vacuole? Fazila Fawaz, you have potassium and chloride. Very good. Okay, it contains potassium and chloride. Pigments, example would be anthocyanin. Okay, those are pigments that are found inside the vacuole. So this is correct. The composition of the cell sap. What is the cell sap? The cytosol. Okay, the cytosol. Yes, the cytosol. And the the cell sap is what is found inside the vacuole. And cytosol is similar. The composition of the cell sap. The cell sap is what is the liquid that is found inside the vacuole. And the cytosol, the fluid nature that is found inside a cell, is similar. That is wrong. Why? We just saw, no? The sugars are present, ions are present, pigments like anthocyanin are present. So if these components are there in the cell sap, inside the vacuole, then they can't be the same as cytosol. Right? So second one is incorrect. Third one, the sap pigments give turgidity and support to the cell. No, it is not the sap pigments. What is turgidity? I told you to imagine a full water bottle, not a metal water bottle, a full plastic water bottle. It's difficult to squeeze or break it because of the water that is present inside, right? So if you take a vacuole, vacuole provides turgidity to the cell due to the water that is present in the central vacuole, not due to anything else. So then this is incorrect. Fourth one, water insoluble pigments such as anthocyanins are present. It will not be present in the cell side if it is insoluble, right? Water soluble pigments such as anthocyanins are present. So then this is incorrect. Ions such as calcium and chloride are present. False. It was potassium and chloride. So then fifth one is also incorrect. Answer is answer number 1. So 10 questions are done. So if right. So up to that point, if you have any questions, please ask. If you have any questions, please ask. Okay, there is a question regarding the cholesterol part. Where is it mentioned that cholesterol provides fluidity? Okay, this is what you mean by reading the fine print. Okay, so when they talk about fluidity in the plasma membrane, okay, you would go to the plasma membrane chapter and read about fluidity. But go to functions of lipids under biological molecules. Second point, maintain the fluidity of plasma membrane, phospholipids and cholesterol. Okay, maintains the fluidity of plasma membrane, phospholipids and cholesterol under functions of biological important lipids. Their functions are given. Second function, maintain the fluidity of plasma membrane, phospholipids and cholesterol. So you have to make sure to integrate information across 2 topics. So just because you study plasma membrane, you can't now. In plasma membrane, they talk about phospholipids, no? So you should not ignore the phospholipids that were talked about in lipids. So both should be combined in your brain, but when they ask you to write a structure of the plasma membrane, just that part is enough. A hormone can be a lipid and a protein, right? Hormone can be lipid and protein, yes. Hormone, there are lipophilic hormones, that is the hormone, now cholesterol. So if you take steroid hormones, testosterone, progesterone, estrogen, corticosteroids. So these are lipid hormones. Proteinous hormones would be insulin, glucagon. Those are protein hormones. Okay. So hormones can be lipid or proteins. But in plasma membrane, cholesterol molecule provides rigidity and stability, right? Yes. So under the plasma membrane note, it says rigidity and stability. Under the lipid note, it says fluidity. So if you take cholesterol, it has 3, no? Rigidity, stability, fluidity. Okay. All right. So what is the function of a condenser in a microscope? Condenser in a microscope is usually found in the bottom part of a microscope. You can control the amount of light that enters through the specimen. So that is the function of a condenser. So if you do not have any other questions, I will move on to the next topic. Okay, right. Question number 11. Select the combination that correctly shows the cellular organelle and its functions. Organelle functions, and they have given this combination part here. So how are you supposed to answer this question? Okay. Write down over here all the functions that can come to this organelle. So we'll take it this way. Rough endoplasmic reticulum is A. Over here, what is the functions of rough endoplasmic reticulum? Synthesizing lipids and steroids, wrong. That comes for smooth endoplasmic reticulum. Production of non-cellular cell wall components, wrong. That comes for Golgi apparatus. Production of vesicles for protein transport, yes, that comes for rough endoplasmic reticulum. Now, does rough endoplasmic reticulum synthesize the proteins? No, it is the ribosomes which are bound to the rough endoplasmic reticulum that synthesizes the proteins. The rough endoplasmic reticulum can modify and package it into these vesicles for protein transport. So AR is the correct combination. What about digestion of worn out organelles? That is something belonging to lysosomes. So A, only R is a correct function. Then let's go with B. Smooth endoplasmic reticulum synthesizing lipids and steroids. So that is correct. Synthesizing lipids and steroids is correct. Production of non-cellulose cell wall components, that belongs to Golgi apparatus. So then that is wrong. Production of vesicles for protein transport. Golgi, smooth endoplasmic reticulum, yes, it does produce vesicles for transport, but transport within the cell. So R cannot come for smooth endoplasmic reticulum. Or can it? Is the protein transport, is it mentioned as protein for smooth endoplasmic reticulum? No, it hasn't mentioned as protein. Okay, it is producing transport vesicles within the cell. So then R also cannot come for smooth endoplasmic reticulum. Then S, digestion of worn out organelles, again, not a function of smooth endoplasmic reticulum, that is a function of the lysosome. So let's go to the C part. Golgi apparatus. Synthesizing lipids and steroids, no, that belongs to smooth endoplasmic reticulum. Production of non-cellulose cell wall components, that is correct. CQ. Production of vesicles for protein transport. Technically, yes, Golgi apparatus. If you remember the function of Golgi apparatus, collecting, packaging, distribution of proteins and membrane lipids. So distribution of these proteins and membrane lipids, they are sent out in vesicles only. So Golgi apparatus, yes, production of vesicles for protein transport is correct. It can be taken as correct. So CQ, CR is also there. Lysosomes, digestion of worn out organelles, DS. So AR, BP, CQ, DS. And that is answer number 1. So do you see how to do this question? When you go in this manner, when you look at these options, sometimes you get confused because you see it's just a bunch of letters. But if you can derive an option by without looking at these options itself, if you look at the organelles and the functions and you match it and try not to match it over here like this. Okay, like we did in grade 4 or something. Don't match it like that. We have to follow it. No? So then you might make a mistake. Now, especially in the A-level exam, when you have so much pressure. Now I've seen some students have sent me messages, okay, quite a few who studied well for the exam. Now forget about the students, even one of my friends back in A-level time in 2021, that person used to get more than me for biology. Okay, I used to get only 42, 43 for the past papers before going to the A-level exam. That person used to get 48, 49. But when the results came, I got an A and that person got a C for biology. The reason is once the person went to the exam, started freezing. It can't recall. So much pressure and all of that, got so nervous in the exam hall. And especially we were in Corona time, no? So that was the first exam that we probably did. Okay, so properly did. So then don't go and start drawing lines like this, which you will have to follow. In that pressure, you might, look at it incorrectly. But if you write it down like this, there is very less chance you will get it wrong. So answer is answer number 1. Okay, right. Now time to ask you the question. Okay, raise your hand if you can tell me all the functions of the rough endoplasmic reticulum. Okay, Vikasha, can you tell me all the functions of the rough endoplasmic? Okay, she put her hand down. Then, who haven't I asked? Suha Fazil, can you tell me all the functions of the rough endoplasmic reticulum?
Facilitate the growth of its own membrane by adding proteins, carbohydrates, phospholipids, synthesizes glycoproteins, synthesizes membrane phospholipids, produce transport vesicles, and transport protein synthesized by bound ribosomes.
Okay, good. Right? So, organelle function, that is 100% a question that is going to come to your A-level exam. 100%. Every single paper had a question on organelle. So, if you can remember the organelles and their functions, you have guaranteed 1 mark in your A-level exam. Very easy also. Please do not miss it. Okay. Question number 12. Which of the following statements are correct regarding the cell junctions found in animal cells? Okay. Tight junctions tightly connect the plasma membrane of adjacent cells, preventing the leakage of extracellular fluid. That is correct. You can find these tight junctions in your skin cells. So how do tight junctions look like? So take a look. You can see the tight junctions here. So these tight junctions are present and they, like a tailor who sews two pieces of cloth together. Likewise, you have these proteins that sews the walls or the plasma membranes of two cells together, such that making sure that nothing can leak through this gap. Okay, so these proteins form continuous seals around the cell to prevent leakage of fluid. So then the first A statement is correct. A statement is correct. B, intermediate filaments in desmosome junctions mechanically connect the cytoskeleton of adjacent cells. So these desmosomes are found in muscle cells. So if you take muscle cells, each cell in the muscle must contract. Okay, so when they are contracting, to prevent them from separating or preventing the cells from separating, you need to have these desmosomes that are present like staplers, which staple two muscle cells together, specifically in the cardiac muscle, such that when it is contracting, you don't separate the muscle cells. Okay, that is why desmosomes are very important. So they involve the cytoskeleton so that it can provide additional support to make sure that this contraction takes place properly. So B is also correct. Then in gap junctions, specific proteins form continuous seals around the cells. We just saw, no? Continuous seals belongs to tight junctions, not gap junctions. In gap junctions, you just have proteins which connect two cells, which can allow ions and other molecules to pass through. Then D, communicating junctions allow the passage of sugars and amino acids between adjacent cells. Communicating junctions, another name is gap junction. So where can you find gap junction? One is animal embryo and the other one, raise your hand if you can tell me the answer. Okay. Sahla Fazil, can you tell me what is the other one which has communicating junction?
Heart muscles.
Heart muscle. Okay, you can't just put muscles, you have to mention cardiac muscles. Okay. So based on that, A, B, and D, correct answer is answer number 4. Right. Select the correct matching response from the following: prophase, prometaphase, anaphase, telophase, and here you have the things that occur during each phase. Okay, let's go from this side. Fragmentation of nuclear envelope. Fragmentation of nuclear envelope takes place in which phase? Anyone can answer the question?
Prometaphase.
Mitosis.
Take a look at this. Can you see? Over here in prometaphase only you will have fragmentation. In prophase, the nuclear envelope looks good. But in prometaphase only the nuclear envelope is fragmenting. Right? So prometaphase is where fragmentation of the nuclear envelope takes place. So BP. Then spindle microtubules get depolymerized. Spindle microtubules get depolymerized. That should take place in which phase?
Telophase.
Telophase, when all the cell division is over, no? At the last part, because in the beginning part you have spindle microtubules which form and they get connected to the chromosomes and then they separate it. But at the end, when the microtubules are done, that is when they get depolymerized or decondensed, okay, or broken down. Okay, so then that takes place in telophase, like you have correctly mentioned. Centrosomes move towards opposite poles, and that has to be prophase. Why? Because why does the centrosome need to move towards the opposite poles? If you take a look at this.
For microtubules.
Yes, very good. If you take a look at this, you can see the centrosomes are present. Over here, you can see that the centrosomes are moving towards opposite poles. Why does it have to move towards opposite pole? So that when it is in the opposite poles, then only it can connect to each chromosome and separate the chromosome. It can connect to each chromosome and separate the chromosome. All right. That is why centrosomes need to move towards opposite poles. But before they move towards the opposite poles, centrosomes must duplicate. Where does the duplication take place? Anyone? Anyone? Duplication, where does the duplication take place?
So can you please repeat the question?
Yeah, duplication of centrosomes, where does it take place?
G2 phase, in the
Interface.
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 G2 phase of interface. Very good. Okay. Right? G2 phase of interface. All right. Cell elongates as non-kinetochore microtubules lengthen. So then that happens in anaphase. Okay, here it is not drawn clearly, but I will show you. So microtubules, you have two types, right? Kinetochore and non-kinetochore. Kinetochore microtubules are the one attached to the chromosomes. Non-kinetochore microtubules are not attached to chromosomes. They interact with the non-kinetochore microtubule of the opposite pole. So what is the purpose? You can imagine as if they are pushing each other. As they are pushing each other. Since they are pushing each other, only this goes in this direction, this goes in this direction, causing it to separate like this. Formation of the cleavage. Okay. Right. So then that happens in which phase? Anaphase. So then the correct answer would be second one. AR, BP, CS, DQ. Is that okay? Question number 13.
So when we write essays on these nuclear divisions, is it necessary to draw these diagrams?
Yes, I have allocated marks for this diagram. Okay. However, most importantly, it is not the diagram, it is the text. Because no matter what essay you take, maximum they would have allocated for the diagram would be 10 marks out of 150. So 140 is from text. So don't worry about if you miss a diagram or not, unless the diagram helps you to write the essay. You can just write the essay and then put the diagram at last if you really need to draw the diagram, because the text is the most important part. After that, you can draw the diagram unless the diagram helps you remember what to write. Okay, so then question number 13 also done. Question number 14. So then this is regarding cancer cells and plant galls. First statement, externally supplied growth factors are always essential for cancer cell division. That is false, because cancer cells can grow uncontrollably. So they either do not need growth factors at all, or they can synthesize their own growth factors and continue the cell division. Okay. To prevent these cancer cells only, we have these cell cycle checkpoints, but cancer cells do not care about these cell cycle checkpoints. Okay. Second one, plant galls are formed due to controllable mitotic division of plant cells. It is uncontrollable mitotic division, no, not controllable mitotic division. Third one, cancer cells can synthesize the growth factors they need by themselves. So like I just mentioned, cancer cells can synthesize their own growth factors, or they do not need growth factors to continue in the cell cycle. Fourth one, galls are formed when there is a proper balance between auxins and cytokinins, not proper balance when this balance is lost. That is when you have gall formation. Benign tumor cells can enter the lymph and spread to other parts of the body. Okay. Raise your hand. I need a review from this from one student. Okay. Okay, Vikasha, can you tell me statement 5, why is it wrong?
Benign tumors stay in the same site.
Very good. Benign tumors stay in the same site. Continue.
Therefore, they do not spread and enter other parts of the body.
Okay, so if it is entering the lymph and spreading to other parts of the body, what do you call that?
Malignant tumor.
Malignant tumor. The process of spreading from one place to another place, what do you call that?
Metastasis.
Metastasis. Very good. Now my question to you, right? Not to you specifically. Let's see who can answer this question. Benign tumors are always harmless. Raise your hand if you can answer that question.
False.
Right. False.
False.
Why is that?
Because
It
they
can
can be
be
safely
removed.
removed by one operation.
Sorry?
They can be safely removed by an operation.
Okay, but my question is benign tumor, can benign tumors be harmful or not?
Yeah, because
Can
they
be
do not
harmful.
cause problems, but they can cause minor problems.
Uh, How can they cause minor problems? Who said that? Sahla, no?
If it is not removed by surgery, it will become a malignant tumor.
Not really. Most of the benign tumors.
Some tumors are large, that will cause issues.
No, not really. No. Anyone still? Still the answer didn't come.
Unless
Not
benign tumor
always removed by surgery.
No.
if
Think
benign
and tell
tumors
me.
don't
We have a
become malignant tumor.
okay. We have one person who told Amira Munsir because of the location. Tell me which location do you think benign tumors will be an issue?
So breast cancer
Brain.
in the breast.
Sir,
Brain, yes, brain. Okay, benign tumors in the brain. They are not harmless, they are harmful because they can compress on the sensitive structures present inside the brain. Okay, if you use the word cancer, it's not a benign tumor anymore. Okay, cancer means it is a malignant tumor. All right. Okay, so what are the modes of spread? Can they spread only by lymph? No, they can spread by blood as well. All right, so question number 14, answer number 3. Then question number 15. All cells synthesize ATP from oxidative phosphorylation. That is wrong, no? You have 3 types of phosphorylation present. You have photophosphorylation, substrate level phosphorylation, and oxidative phosphorylation. So substrate level phosphorylation only you have glycolysis, then you will have citric acid cycle. Oxidative phosphorylation is the electron transport chain. Photophosphorylation is the light dependent reaction in photosynthesis. So all cells synthesize ATP from oxidative phosphorylation will be false. Why? For you to have oxidative phosphorylation, what do you need? You need a mitochondria. Or you can also have internal membranes. Okay, or internal membranes. But again, think about it. If you use oxygen only, you can have oxidative phosphorylation. If you use oxygen only, you can have oxidative phosphorylation. So if you are using oxygen, then it has to be an aerobic respiration. So now can you remember, are all organisms aerobic? No, there are some organisms which are only anaerobic. So then these organisms don't have oxidative phosphorylation. So therefore, saying that all cells synthesize ATP from oxidative phosphorylation is false because anaerobic organisms do not have oxidative phosphorylation. So then first one is wrong. Second one, ATP binds to the same enzyme as an inhibitor and slows down catabolism during allosteric regulation. So what is catabolism? Catabolism is breaking down of large molecules into smaller, simpler molecules. So this releases energy and that energy is used to synthesize ATP. So when you have a lot of ATP, when you already have a lot of ATP, you don't need to produce more ATP. No, if you already have a lot of ATP, you need to reduce the amount of ATP. That is when ATP itself can go and act on this catabolic pathway to inhibit it. Because if you have a lot of ATP and you still keep producing a lot of ATP, it is a waste. Okay, that is why ATP goes and inhibits the catabolic pathway. So second one is correct. Third one, when ATP is oxidized, free energy yield is minus 30.5 kilojoules per mole. Okay, time to ask you all. Raise your hands if you can give me a review for the third statement. Fatima Amana, can you tell me?
Not during oxidized, it's during hydrolyzing.
Very, very good. It is not oxidized, it is hydrolyzed. Okay, when ATP is hydrolyzed. What is the reaction? ATP plus water will give you ADP plus phosphate, no? And the free energy yield is, yes, it is minus 30.5 kilojoules per mole. Then, fourth one. Fourth one, most biological reactions use the energy released during breaking of the bond between the pentose sugar and the phosphate group of ATP. So let me draw the structure of ATP. Here you have a pentose sugar. Here you have a nitrogenous base. Here you have a phosphate, phosphate, phosphate. Right? For you to get the energy that is released, from ATP hydrolysis, it is actually this terminal phosphate that breaks down. It is the terminal phosphate that breaks down. So in this statement, what are they saying? Energy released during breaking of the bond between the pentose sugar and the phosphate group. Pentose sugar and the phosphate group means this one. This is not the bond that breaks down during ATP hydrolysis. It is this bond that breaks down during ATP hydrolysis. So then this is incorrect. Okay, ATP is a nucleoside. It is wrong, right? It is a nucleotide. A nucleoside means a nucleotide without a phosphate group. Can you see there are 3 phosphate groups? So then this is opposite of a nucleoside. Okay, so then here you have adenine, and this one, is it a deoxyribose sugar or ribose sugar? Anyone can answer?
Ribose sugar.
Ribose sugar.
Okay, right. So correct answer here would be answer number 2. Okay, so then that is half that is done. All right. When ATP hydrolyzes, it yields minus 30.5 kilojoules of amount of free energy. Yeah. Okay, that is correct. It's hydrolysis. Because it can develop into a malignant tumor if not treated. Yes, that is also true. Benign tumors can develop into malignant tumor. But if it doesn't become malignant, a benign tumor alone can be an issue because it can, in a sensitive area like the brain, it can cause damage. So we are halfway there, 15 out of 30 MCQs. Is there anyone who has attempted this paper from, anyone who has attempted this paper not from the 2028 batch? Raise your hand. Right. Can you send me in the privately in the chat itself how much you got out of 15? I just want to know. $14 out of 15. Good, not bad. Okay. Question number 16. Right. The most accurate statement regarding the mechanism of enzyme action is enzymes are not always fully specific to the substrate. Why is this wrong? Anyone who can answer that question? Raise your hand.
Enzymes are complementary to their substrate, but there are certain enzymes that can slightly change their shape of the active site because they are not rigid structures, which we call as induced fit mechanism.
Okay. So then saying that they are not always fully specific to the substrate is okay, no? Because like you mentioned, some need induced fit means then enzymes are not always fully specific means that should be correct, no? So then why is that wrong, Amnaza?
They can only make a slight change, so we say enzymes are not fully specific to the substrate is wrong.
Okay. We'll ask others also.
So they are specific, but they are not fully complementary to the substrate.
Yeah, there you go. Right? Enzymes are not always fully specific is wrong. Enzymes are highly specific. They are fully specific. Yes. They are highly specific to the substrate. Okay. They are not always fully complementary. You need to understand the difference between complementary and specific. Okay, complementary and specific. It might not be enough. For example, if you take this as the active site and this as the substrate, you can see it is not fully complementary, right? But this active site can only bind with this substrate. Yes, it is highly specific, but it is not fully complementary. Once it comes close together due to interactions, it can eventually be fully complementary. Okay, it is highly specific, but not always fully complementary. That's why you have the induced fit mechanism. Okay, so the active site of an enzyme is fully complementary to the shape of its specific substrate. That is false because I just told you it is not always fully complementary. Third one, protein components essential for the catalytic action of some enzymes are called cofactors. That is false. For it to be a cofactor, it needs to be non-proteinous components. Non-proteinous components. Because if it is a protein component, enzymes are also proteins. So then why can't we just assume that these cofactors are coenzymes are part of the enzyme. The reason why we talk about them differently is because they are not proteins. Okay, right. Few amino acids form the active site of the enzyme. That is true. Few amino acids form the active site. The remaining amino acid forms the rest of the enzyme, which maintains the shape of the active site. Then reversible cofactors are called coenzymes. No, organic cofactors only called as coenzymes. Organic cofactors. Organic cofactors are called as coenzymes. Okay. Question number 17. The incorrect statement regarding the enzyme inhibitors is. The incorrect statement regarding the enzyme inhibitors is. Most of the company, now before we even go to the question, underline this. Incorrect state. They are not asking the correct statement, they are asking the incorrect state. Most of the competitive inhibitors are non-reversible inhibitors. False. Most of the competitive inhibitors are reversible inhibitors. Now, if you can answer my question, please raise your hand. How will you reverse a competitive inhibitor? Raise your hand if you can answer the question. Okay. Sahla Fazin, can you tell me?
So can you repeat the question?
My question is, how can you reverse a competitive inhibitor?
By increasing the substrate concentration?
Very good. By increasing the substrate concentration only, you can reverse a competitive inhibitor. So saying most of the competitive inhibitors are non-reversible is incorrect. However, here they are asking you the incorrect statement. So then it is correct. Molecules or ions are enzyme inhibitors. Yes, certain molecules and ions are enzyme inhibitors. That is directly from the textbook. Drugs used against microbes are reversible inhibitors. That is true, but you need to understand this is just for A levels they have used like this. But once you come further, you will know drugs used against microbes are both reversible inhibitors and irreversible inhibitors. You need both. But for now, for your exams, just write it as reversible inhibitors. Okay. Fourth one, competitive inhibitors compete with the substrate for the active site of enzyme. Yes. That is why when we increase the substrate concentration, we can overcome the competitive inhibition. Then fifth one, non-competitive inhibitors change the shape of the active site. That is true. So how do non-competitive inhibitors work? Non-competitive inhibitors, another molecule goes and binds to another site, which is not the active site, which causes the active site's shape to change. So that is non-competitive inhibitor. So correct answer here would be answer number 1. Can you all hear me?
Yes, sir.
Yes, sir.
Is the board clear?
It's clear, but like in the middle, it used to get stuck, and you're also getting stuck.
Okay.
So we can't
Yes,
hear
sir.
properly.
All right. Sorry about that, but I think it should be okay now. So what topics will be questioned for the first paper we have on August 31st? Okay, this is for the 2027 paper class, not for the 2028. So you all can close your ears. The first paper is testing on the first term, which would be from the beginning of Unit 1 until Kingdom Protista in Unit 3. Okay, up to the end of Protista in Unit 3. That would be the first term. Certain papers have only Unit 1 and Unit 2. Certain papers go all the way till Kingdom Protista. So anyways, be thorough with the first term. And then you can attend the paper on August 31st. While the 2028 batch, you all don't come for the paper class, okay? You all come next year because I'm planning on giving the same papers because I have made almost around 40 something papers. So I can't be making 40 papers every year, no? So please don't come. Okay. Right. Question number 17 is discussed, right? Yeah. So we move on to question number 18. Regarding the regulation of enzyme activity within cells, the correct statement is allosteric enzymes are often composed of several subunits, and the binding of a regulatory molecule to the active site of a subunit changes the shape of the entire enzyme complex. Okay, I'm not sure why that is wrong. Anyone, can you raise your hand if you can give me the review for the first one? Okay, Naziha, can you tell me why the first one is wrong?
Regulatory molecules don't bind to the active site.
Where do they bind to?
To the regulatory site, often located where the subunits join.
Yes, very good. They are bound, they are binding to allosteric site or the regulatory site. And where do you find this regulatory site? Often where the subunits join. So now, did you notice one thing in the way that she asked the question, answered the question? So when I asked what is wrong with this statement, instead of just saying that, regulatory molecule does not bind to an active site, she said regulatory molecule does not bind to an active site. It binds to a regulatory site or allosteric site, which is usually found where other subunits join. That is how you're supposed to answer the question. Okay, not it will help you now also, but mainly it will help you in the future. Now, even when you write an essay answer, that is why most people struggle with essays, because you don't know what to write next. So you should always try to answer in such a way, you have to imagine that the person who is marking the paper doesn't know anything. So you need to go from the beginning, tell him or tell her, this is how this process occurs, A to Z, exactly what happens before, what is happening currently, what happens after, what happens if it didn't happen, what happens if that happens, all the things you should write properly, what is there in the resource book. Okay, nothing is not important. Then question number 2, statement number 2, in cooperativity, the binding of one substrate molecule to an active site stimulates or inhibits the activity of other active sites. Why is this wrong? Raise your hand if you can answer that question. Let's go with Pranati. Can you tell me why second is wrong?
It does not inhibit, it only stimulates.
Cooperativity is only about stimulation. Okay, it does not inhibit. Binding of a substrate onto an active site will increase the binding of all the other active sites. Okay, not inhibiting, never inhibiting. So that is why the second one is incorrect. Third one, when the ATP concentration in the cell increases, ATP acts as an allosteric inhibitor for those metabolic enzymes, reducing the catabolic rate. So this is similar to the enzyme question that we saw before, allosteric enzyme ATP question we saw before. So third one is the correct answer here. Fourth one, in feedback inhibition, the intermediate of a metabolic pathway binds to the active site of an enzyme in an early step of the pathway, stopping the process. Feedback inhibition does not work using intermediate. It is the end product. The end product is what goes and inhibits the pathway, not the intermediate. Okay, so this has to be end product. Then fifth one, allosteric enzymes are often composed of a single polypeptide chain and have multiple allosteric regulatory sites. That is false. Allosteric enzymes, they are mainly composed of more than one subunit. But now can you tell me, okay, allosteric enzymes can never be a single subunit enzyme. True or false?
False.
False, because they are usually or most often more than one subunit. So if they are most often more than one subunit, what does that tell you? Rarely they can be a single subunit enzyme also. Okay. So can we say that they are metabolic enzymes? The third answer, yes, we can say that they are metabolic enzymes. What does metabolic enzyme mean? Metabolic means chemical reaction. It includes catabolic and anabolic reactions. Okay. So metabolic includes anabolic and catabolic. So here they say metabolic and they say reducing the catabolic rate. So they are specifying the metabolism that they're talking about here is the catabolism. Okay. If they have mentioned metabolic reducing the anabolic rate, then you can argue. But here they say metabolism reducing the catabolic rate. So that is why third statement is correct. So then the correct answer here is 3. Why everything is wrong, we just discussed it. So we can go to question number 19. Right. Can you all hear me and see me clearly now?
Right
sir.
now can, sir. But in the earlier question when you were explaining, you were completely stuck.
Right. So which part should I repeat that? Which part was I stuck in?
I think it was a fourth and fifth answer discussion.
All right. So I will repeat the fourth and fifth once more. So in feedback inhibition, the intermediate, it is not the intermediate, it is the end product of a metabolic pathway that binds to the active site. Okay, not the intermediate. Then allosteric enzymes are often composed of a single polypeptide chain. No, they are often composed of more than one subunit. Okay, they can be one subunit rarely, but often they are more than one subunit. Okay, that is why the answer is 3 here. Right. Then question number 19. Which of the statements is correct regarding the linear electron flow of photosynthesis? First one, splitting of water takes place at P700 of photosystem 2. In photosystem 2, do you have P700 or P680?
P680.
You have P680, right? Splitting of water takes place at P680 of photosystem 2, not P700. Second one, excited electrons of PS2 is used to neutralize the excited electron of PS1. So can you see here? The excited electron in PS2 will neutralize the excited one in PS1. Okay, neutralize the excited PS1. Then question number 3, NADPH is reduced and yields NADP plus catalyzed by the enzyme NADP plus reductase. That is false, no? NADPH is reduced and yields NADP plus is the incorrect part here. NADP plus only will be reduced to form NADPH and the enzyme is NADP plus reductase. Okay, so third one is incorrect. Fourth one, electrons released as a result of hydrolysis are used to neutralize the excited photosystem 1. So splitting of water takes place in photosystem 2, not in photosystem 1. Photosystem 1, the photosystem 1 is neutralized by the electrons from photosystem 2. Photosystem 2 is neutralized by the electrons from water. Okay. Fifth one, utilization of ATP is due to the passage of electrons from a higher energy state to a lower energy state. So when the electrons go from a higher energy state to a lower energy state, is ATP being utilized or is ATP being produced?
ATP is produced.
ATP is being produced. Okay, that is why fifth statement is incorrect. Okay, right. So question number 20. The correct statement about photosynthesis is, in excess light, carotene and xanthophyll produce oxidative molecules that are harmful to the cell. So is that correct? No. In excess light, these carotene and xanthophyll are the ones that provide the photoprotection. Okay, carotenoids provide the photoprotection to prevent the formation of these oxidative molecules. So then the first statement is incorrect. Is that clear?
Yes, sir.
Yes, sir.
Yes, sir.
In C3 plants, the ATP produced in the light reaction is used only for the formation of 1,3-bisphosphoglycerate and for RUBP regeneration. Okay, so formation of 1,3-bisphosphoglycerate is carbon fixation. Yes or no?
Yes.
Yes or no?
Yes, sir.
What happens
Yes,
in
sir.
carbon fixation? 6-carbon compound will break down into 2 3-carbon compounds, PGA. That PGA will convert to 1,3-bisphosphoglycerate. Then 1,3-bisphosphoglycerate becomes what?
Glyceraldehyde 3-phosphate.
Glyceraldehyde
GGP.
3-phosphate. And that happens in which phase? Reduction. And then once the glyceraldehyde 3-phosphate is sent out, you have the
Reduction.
regeneration of RUBP. Okay, so third phase is regeneration of RUBP. So now carbon fixation ATP is required. Regeneration of RUBP ATP is required. For reduction, what is required additionally?
Phosphate
NAD.
group.
NADPH. You need NADPH. Now for reduction, do you need ATP?
According to the textbook, it's yes.
Yes, very good. According to the textbook, yes, for reduction, you need ATP. But in real life, you do not need ATP for reduction. Reduction is done only by NADPH. That is why this question, all is the answer based on the resource book. In C3 plants, ATP produced in the light reaction is used only for the formation of 1,3-bisphosphoglycerate. That is carbon fixation and for RUBP regeneration. No. According to the resource book, you can see ATP is also necessary for the formation of glyceraldehyde 3-phosphate. But like I told you when you did the theory class, however, in real life, glyceraldehyde 3-phosphate formation, only NADPH is required. But in our textbook, they use both NADPH and ATP. That is why this question you don't have an answer, because according to the resource book, this is wrong. Then third statement, in C4 plants, the leaf mesophyll cells contain chloroplasts, which are relatively large, while in bundle sheath cells contain only PS2. So we will come to this diagram over here. So bundle sheath cells, you only have PS2. Can anyone tell me what happens at PS2? Anyone?
Linear electron flow.
Sorry? Linear electron flow. What does it produce in PS2?
ATP and NADPH.
PS2, what is produced?
ATP.
ATP.
So that is
ATP.
electron flow, no ATP is produced from
Cyclic electrons.
From PS2, the thing has to go to PS1. What happens at PS2 alone? What is released as a byproduct?
Splitting
ATP.
of water.
Yes, splitting of water. What happens? What is produced in the splitting of water?
Oxygen, hydrogen ions, and electrons.
Right. Now the C4 pathway, the purpose of having division of labor between mesophyll cells and bundle sheath cells, it is so that oxygen doesn't come into contact with the Rubisco of this Calvin cycle. Okay, oxygen doesn't come into contact with the Rubisco of this Calvin cycle. Okay, so that if this bundle sheath cell chloroplast has PS2, it will generate oxygen. Okay, so that is why this is wrong. Bundle sheath cells cannot have photosystem 2. So bundle sheath cells cannot have photosystem 2. Fourth one, the rate of photosynthesis depends on the factor that is most available at a given time. Is it correct or wrong?
Wrong.
Wrong. It depends on the factor that is the least available at a given time, because that is the who is the one that came up with this theory?
Blackman.
Blackman.
Yes, Blackman. Okay, limiting factor. So then we have a question, is it NADP plus reductase or NADP reductase? Is NADP plus reductase, okay? NADP plus reductase. All right. Grass, rice, and barley are examples for C3 plants. Wheat, rice, and barley are examples for C3 plants. Grass, sugarcane, and maize are examples for C4 plants. So then this is also incorrect. So now you understand why question number 20 I have given all. Okay, so we have completed 20 questions. We have 10 more questions more. So the ones who have done the paper just up to now, can you tell me out of 20, how much have you gotten? Right. Okay, question number 21. Am I lagging?
Yes, sir. You are now okay.
Yes.
Now, okay, sir.
Oh, okay. Right. So no worries. Today I'm planning to go and buy that cable which can directly connect the router with the computer. So there will not be any problems hereafter. Okay. So today only I got a chance to have a free day because it's some poya day, no? Day. Right. Question number 21. The intermediate compound that is formed in the Salvin cycle as well as the anaerobic respiration. This is actually such a simple question. Even though I think everybody got this question, the highest was 29, right? I think the person who got 29 got this question wrong. Even though this question might look complicated, it's very simple. The intermediate compound formed in Calvin cycle as well as the anaerobic respiration. So anaerobic respiration, what happens first? You have glycolysis. And then you have the two types of fermentation, ethyl alcohol fermentation or lactic acid fermentation. So the common for anaerobic respiration is glycolysis. So then this question is asking what is common to glycolysis and Calvin cycle? That's all. Ethanol. Ethanol is not even common in anaerobic respiration. It is only common in ethyl alcohol fermentation, not in lactic acid. So first one is anyway wrong. Acetaldehyde also same thing. It is only found in ethyl alcohol fermentation, acetaldehyde, not in lactic acid. Malate. Malate, is it even found in the Calvin cycle, malate?
No.
No, it is found in the C4 pathway. So then this is wrong. Pyruvate, is it found in the Calvin cycle even?
No.
No. So if you just know what is the thing that is actually found in the Calvin cycle, you can come to the correct answer, G3P. So then your next question would be, what does G3P have to do with anything with glycolysis? So this is why if you had looked at the last diagram in the tube, which is that, how different respiratory substrates are used up, you can see carbohydrates. Glycolysis, you have glucose, which converts into glyceraldehyde 3-phosphate, which becomes pyruvate. So glyceraldehyde 3-phosphate is an intermediate in glycolysis. And glyceraldehyde 3-phosphate is also found in Calvin cycle. So correct answer would be answer number 3. Right. Then,
Sir, can't
Yeah.
we take pyruvate? In C4 pathway, pyruvate is also related, right?
but we are talking about the Calvin cycle, no? Not C4 pathway. Oh, it's the wrong one. We are talking about the C4 pathway, no? Not pyruvate. All right. So then the next question number 22. Under normal conditions, which of the following are the major limiting factor? So what is the major limiting factor under normal conditions? It is carbon dioxide concentration, right? When the 6-carbon product breaks down, doesn't it form the 2,3-carbon pyruvate? So that is in glycolysis, no? It forms pyruvate. But in Calvin cycle, you don't have pyruvate, right? In Calvin cycle, you have 6-carbon, which becomes 3-PGA, which becomes 1,3-bisphosphoglycerate, which becomes glyceraldehyde 3-phosphate, which becomes RUBP. So there's no pyruvate in the Calvin cycle, but there is a pyruvate in the glycolysis. So glucose, glyceraldehyde 3-phosphate, which becomes pyruvate. Okay. Carbon dioxide concentration. So all of you all mentioned this. Now to overcome this issue with a limiting factor, is there any technology present? Raise your hand if you know the answer. Okay, Naziha, can you tell me?
Concentrating carbon dioxide.
Yeah.
Example, tomatoes are grown in carbon dioxide enriched environment in greenhouses.
Very good. Carbon dioxide enriched environment in greenhouses. Okay. 22nd is answer number 2. All right. Then question number 23. 23. Production of energy within a cell is divided as follows: photophosphorylation, substrate level phosphorylation, and oxidative phosphorylation. Which of the following methods are involved in ATP during cellular respiration? Okay. So photophosphorylation, can it take place in the cellular respiration?
No.
No.
It only takes place in light dependent reaction, linear electron flow and cyclic electron flow. So then this is incorrect. Substrate phosphorylation,
Yes.
yes, it takes place in glycolysis and it takes place in the citric acid cycle. Okay, glycolysis and citric acid cycle. Oxidative phosphorylation, like we all know, it takes place in the electron transport chain, which is the last step. So then the correct answer here would be B and C. Answer number 5. Pretty easy. Then question number 24. Which of the following does not take place in the citric acid cycle? Carbon dioxide is released from decarboxylation. Is that true or false?
True.
True. How many carbon dioxide is released per cycle?
1.
Two.
2.
Two carbon dioxides are released per cycle. Okay. Regeneration of citrate from oxaloacetate, right? Which of the following does not take place, no? So regeneration of citrate from oxaloacetate. So is that what happens?
No.
You have the regeneration of oxaloacetate from citrate.
From citric acid.
Yeah, for you to form citrate, oxaloacetate must combine with acetyl and then only it can form citrate. Okay, so this does not take place. Regeneration of citrate from oxaloacetate. It is actually regeneration of oxaloacetate from citrate. Then third one, FADH2 is generated from oxidation. Is that true?
True.
Is that true or false?
Yeah.
Okay, tell me why. Okay, nice.
True.
Yeah, why is it true?
Because FADH2 and NADH are produced, right?
Yeah, so what does it mean by generated from oxidation? Okay, let me explain. So when you take FAD, right? FAD becomes FADH2, no? So FAD has undergone what? Oxidation or reduction?
Reduction.
Reduction. So if FAD has undergone reduction, then something in the citric acid cycle must have undergone
Oxidation.
Oxidation.
So FADH2 is generated from oxidation. Is that true or false?
True.
True. Okay. Right. Then fourth one, substrate level phosphorylation takes place. So is there a process where ATP is formed directly? How many ATP is formed in citric acid cycle?
1 ATP per citric acid cycle.
1 ATP per citric acid cycle. Fifth one, occurs in the matrix of mitochondria. Where does the citric acid cycle take place?
The matrix of
Matrix of mitochondria.
So correct answer is answer number 2. Right. Question number 25. Select the correct statement regarding anaerobic respiration. Okay. Some bacteria do ethyl alcohol fermentation. It is not some bacteria, it is many bacteria does ethyl alcohol fermentation. Limitation of NAD plus increases recycling of NADH. Limitation of NAD plus increases recycling of NADH and utilization of NAD plus. No. You need to recycle NAD plus. The limitation of NAD plus, you need to recycle NAD plus. Here they say recycling of NADH and utilizing NAD plus. Why is NAD plus even necessary? Because when you take glycolysis, for glycolysis to take place, you need NAD plus. For glycolysis to take place, you need NAD plus. So when anaerobic oxidation is taking place, this NAD plus in glycolysis is converted to NADH. But when there is no oxygen, there is no purpose of this NADH. That is why in fermentation, ethyl alcohol fermentation or lactic acid fermentation, this NADH is recycled to give rise to NAD plus so that glycolysis can take place properly. So that's why they say here, limitation of NAD plus increases recycling of NAD plus, not NADH. Right. Then question number 3. Lactic acid fermentation is a process that produces ATP in the presence as well as absence of oxygen. No, lactic acid fermentation is anaerobic respiration. Anaerobic respiration is in the absence of oxygen. Fourth one, in ethyl alcohol fermentation process, the final electron acceptor is acetaldehyde. Yes, so then you might ask, is it with the final hydrogen acceptor? Yes, hydrogen plus electrons. These electron acceptors or hydrogen acceptors, they accept this hydrogen and electron. So in ethyl alcohol fermentation, the final electron acceptor or the final hydrogen acceptor is acetaldehyde, while in lactic acid fermentation, the final hydrogen acceptor or electron acceptor is pyruvate. Okay. Fifth one, the ratio of the volume of oxygen consumed to the volume of carbon dioxide released for a proteinaceous substrate is 0.7. No, the respiratory quotient for a proteinaceous substrate is 0.8. 0.7, the respiratory quotient is lipids. Okay. Right. So why is statement 2 wrong? Because it recycles NADH and only produce NAD plus, right? Yes, it recycles NADH and produces NAD plus. But here they use the word utilization. What does utilization means? Using NAD plus. Okay, it should be recycling of NADH and production of NAD plus, not utilization of NAD plus.
Sir,
Yeah.
the fourth statement according to resource book is wrong, right?
Why?
Since it's mentioned as hydrogen acceptor.
Yeah, like I mentioned, hydrogen acceptor and electron acceptor both are same. Okay? Because they accept hydrogen and electrons. So that's why final hydrogen acceptor and final electron acceptor, they are referring to the same thing. So statement number 4 is correct. Right. Question number 1 to 25 is done. That is where most people will get all correct or one wrong. But from 26 to 30, that means the multiple response questions, that is A, B, C, D, E, that is where most people make mistakes. Okay? Right. So we have 5 more questions to discuss in the MCQ. So before we move on to the multiple response question, can you let me know if you have any doubts?
Sir,
Yes.
sir, with age, won't the tight junctions degrade?
With age, won't the tight junctions degrade? Why do you say that?
I mean, the heart is pumping continuously, and like, will it give up or like, will it get weak?
Yes, it gets weak, but the weakness is due to something else actually. Due to the proteins that are present. But yes, as you grow old, the proteins do degrade. So it could be tight junctions or any junction. Anything degrades as time passes.
Thank you, sir.
Okay. Let's see, were there any other questions? So can you explain 25 second option once again? Okay. Second option, limitation of NAD plus increases recycling of NADH and utilization of NAD plus. So like I told you, for glycolysis to take place, you need NAD plus. For glycolysis to take place, you need NAD plus. Okay, for glycolysis to take place, you need NAD plus. So when glycolysis takes place, this NAD plus gets converted to NADH. This NAD plus gets converted to NADH. This NADH has some effect or help in the oxidative phosphorylation, but in anaerobic respiration, NADH doesn't have a role, right? That is why for glycolysis, that only through glycolysis no ATP is produced. To regenerate this NAD plus, this NADH must become NAD plus again. So this NADH will donate its hydrogen and its electron. In the case of lactic acid fermentation, it will donate its hydrogen and electron to pyruvate. In the case of ethyl alcohol fermentation, it will donate its hydrogen and electron to acetaldehyde. So when it donates its hydrogen and electron, it becomes NAD plus again. So if it becomes NAD plus again, it can help glycolysis to continuously occur. So NAD plus is important. But here they say limitation of NAD plus increases recycling of NADH. No, it should be recycling of NAD plus and utilization of NAD plus. No, it should be utilization of NADH to recycle NAD plus. That is why the second statement is wrong. So since answer 4 of 25 is only partially correct according to the resource book, in that case, even answer 1 is partially correct according to the resource book. So you can give all since answer is not present according to resource book. Unfortunately, no. Because final electron acceptor and hydrogen acceptor mean the same thing. Why? Because NADH becomes NAD plus means it is undergoing oxidation. So something is undergoing reduction. So when we were explaining this multiple times, we mentioned that whenever they accept electrons, it always hydrogen plus electrons. Whether it is NAD plus, whether it is NADP plus, whether it is FAD, everybody donates hydrogen plus electrons, everybody accepts hydrogen plus electron. So saying it is the electron acceptor is the same thing, hydrogen acceptor also means the same thing. So fourth one is the correct answer. First one, it has to be many bacteria because I can't give first one is the answer. And no, I can't give all as well. The answer is answer number 4. All right. So question number 26. Which of the following statements are true regarding organic molecules? Hemoglobin is a storage protein involved in storing oxygen and carbon dioxide. Is it? Is hemoglobin a storage protein? No, hemoglobin is a transport protein. And it is involved in transporting oxygen and carbon dioxide. So false. A central asymmetric carbon is only present in glycine. That is false because in glycine
It is not present in glycine.
Asymmetric carbon because glycine, the R group is replaced by H. So you do not have 4 different groups. Then one complete turn in the DNA double helix has 20 nitrogenous bases. That is true. Why? Because one turn in the DNA double helix has 10 base pairs. So 10 base pairs means 20 nitrogenous bases. rRNA is the most abundant type of RNA. That is true. Why? rRNA is found in ribosomes and ribosomes are found all over the cell. They are very, very important. Okay. E, triacid glycerol is an amphipathic molecule. No, triacid glycerol is a hydrophobic molecule. Amphipathic molecule is phospholipid. Okay, so answer is C and D, which is answer number 4. Pretty straightforward question. So we will see if there are any questions. So what if it was mentioned symmetric carbon is present only in glycine? Is it that? Yes, because it is not an asymmetric carbon. It's a symmetric carbon. All right, then, okay. I can't stand for long periods. Recently, I went on a trip to Anuradhapura. I think recently means last Friday, Saturday, Sunday. So then I had to walk 32,000 steps in slippers. That's around 20 something kilometers and my feet are completely gone. I'm leaning on one feet when walking. So what if it was mentioned symmetric carbon is present only in glycine? Is it correct? Yes, no, because only glycine has R group replaced as hydrogen. No, everything else has asymmetric carbon. Okay, question number 27. Anyone who got this right, please raise your hand because I tried to play a, I made this question, this part. Okay, not bad, not bad. Because once I come to that point, I will tell you what was the trick I tried to play. Which of the following is an inaccurate comparison? Okay, inaccurate comparison between light microscope and electron microscope. Okay, so they asked the wrong comparison. Glass lenses are used to focus the light rays in light microscope. That is true. You have the objective eyepiece and the objective piece. Both are glass. Powerful magnets are used to focus beams of electrons. That is also true. Image is directly detected by the naked eye. True. Through the eyepiece, you can see the image. Electron micrographs are used. Yes, the computer will generate the image and put it on the screen and we can look at the screen. Plant cells can be observed in light microscope. That is true because living cells can be observed. Plant cells cannot be observed in electron microscope. Why is that? Is this correct or wrong?
They can be observed, but they will die.
Sorry?
They can be observed, but they will die eventually because of the electron.
Dead plant cells can be observed.
Plant cells can be observed. Dead plant cells can be observed. Okay. So now this the trick I played here is this part. I think at least one fellow fell for it. Okay. Because I was sent a message asking why this is wrong. If you take a look in the textbook, it says under light microscope, living cells can be observed and living and non-living can be observed in light microscope, while in electron microscope, it says only non-living. So when I put plant cells, you would have assumed it's a living cell, no? But dead plant cell is also a non-living cell. So dead plant cells can be observed. Dead animal cells can be observed. Any living thing can be observed, but once you put it into the electron microscope, by the process of sample preparation, when you have to prepare a sample to put it into the microscope, it is dead. Okay. So dead cells can be observed by electron microscope. It could be a dead plant cell, it could be a dead animal cell, it could be a dead bacteria, it could be a dead prokaryote, whatever. So this is why this is wrong. Plant cells can be observed, not cannot be observed. Okay. So plant cells can be observed in light microscope because you can put both living cell. Now let's take an epidermal peel. Okay, let's take an epidermal cell. An epidermal cell can be observed through a light microscope. That same epidermal cell can be observed through an electron microscope. Thing is, by the time you do all the processing, that epidermal cell would have been dead and gone by that time. Okay, a dead epidermal cell can be observed through an electron microscope. So I hope you understood that now. Then light microscope, the wavelength of the medium is smaller. Electron microscope, the wavelength of the medium is larger. So what is the relationship? Resolution is directly proportional to 1 over wavelength. Higher the wavelength, lower the resolution. Electron microscope has the highest resolution. So if resolution is high, wavelength is low. So electron microscope should have a lower wavelength. Light microscope should have a higher wavelength. So then this is also incorrect. Then light microscope, dyes are used to stain the object. True. In electron microscope, heavy metals are used to stain the object. Gold also can be used to stain the object. So 27 answer is answer number 4. Is that okay?
Yes, sir.
Yes, sir.
Sir, can you explain the E point again?
Yeah. Dyes are used to stain the object. In light microscope, if you remember, when we watched that video, we used a stain called methylene blue. Right? So when you put a stain, it can stain the cellular structures more in light microscope. Like it can make the nucleus more visible. It can make the cell walls more visible. So when you look at it through a light microscope, you'll be able to clearly see the cell wall, the nucleus, those substances because you added the dye. If not, you're just using water, no? So it will not, if you can see, yes, but it will not be very clear. So dyes can be used to stain the object. It is not a must. It is not a must. Dyes can be used to stain the object to give you a better view, to increase the prominence of the nucleus. Okay, you can see it has properly stained the nucleus and the nucleus is present. It is present centrally, it is present peripherally. All of those things can be seen clearly when you put a dye, but a dye is not necessary. In electron microscope, heavy metals are coating the specimen. So in the case of scanning electron microscope, we used gold. So these heavy metals can dye or stain the specimen, which is necessary to view through an electron microscope. Okay.
Okay, sir.
Right. So can you explain the D part again? When was that message sent? Right now. Okay. So the D part is a wavelength, no? Do you remember what is the resolution? The resolution is how close two points can come so that you can distinguish it as two separate points. Resolution is the issue with microscope. When you have high resolution, you can magnify something properly. So resolution, no, best example would be if you take an unclear photo, no matter how much you zoom in on the photo, you will not be able to see it clearly. So resolution is important and then only magnification. So resolution is dependent on wavelength. Inversely proportional to wavelength. Wavelength of what? Wavelength of the medium. So in light microscope, you're using light rays. In electron microscope, you're using electron waves. Okay. So if you take the wavelength and the resolution, if you want to have maximum resolution or very high resolution, the wavelength should be very low. If you have a very high wavelength, the resolution will be very low. So when you take light microscope, which uses light beams or light waves, the wavelength of light is higher than the wavelength of electron waves. So the wavelength of electron waves are low. So when the wavelength is low, the resolution is high. The wavelength of light waves are higher than electron. So when the wavelength is high, the resolution is low. Okay, that is why we say the wavelength of the medium in electron microscope is shorter and the wavelength of medium in light microscope is higher. Is that okay? Right. So question number 28. Select the correct combination. Anton von Leeuwenhoek, first to describe and record single-celled organisms. That was pretty direct. Matthias Schleider, animal tissues are made up of cells. Do you remember the technique I used? I told you to use to remember who came up with?
Yes, sir.
Yes. Animal tissue is done by Schwann. Swan is an animal. Okay, so this is wrong and this is wrong. Because plant tissues only was by Matthias Schleider. Rudolf Virchow, all cells arise from pre-existing cells. That is true. He contributed to the cell theory. Blackman proposed the idea of limiting factors. That is also true. So correct answer is A, C, and D. Answer number 2. That is also pretty simple. Which of the following phases have cell cycle checkpoints? So which of the phases have cell cycle checkpoints? G1 phase, G2 phase, and M phase. Only these three have cell cycle checkpoints. So what is a cell cycle checkpoint? If you don't go past this cell cycle checkpoint, what happens?
The cell will go into the non-dividing stage.
Dividing stage called the G0 phase.
Most of the cells in our body, are they actively replicating or the G0 stage? Okay. Most of the cells in our body are present in the G0 stage. Okay. So there's nothing you have to learn much about the cell cycle checkpoints. ACD is the correct answer, which is answer number 2. Then last one. Question number 30. Which of the following statements are correct regarding the C4 photosynthetic pathway? Grana in the chloroplasts of bundle sheath cells are very well developed and PS2 is active in them. So bundle sheath cells, if you have PS2, what is generated? Sorry? What is generated?
ATP. Oxygen.
Oxygen, right. Oxygen is generated. In bundle sheath cells, if you have a lot of oxygen, is it good or bad?
Bad.
Bad, because you are trying so hard to send only carbon dioxide. Okay, so this is wrong. Oxaloacetate produced in the mesophyll cells diffuses directly into the bundle sheath cells and enters the Calvin cycle. Oxaloacetate produced in the mesophyll cells. Can you see here? Is oxaloacetate directly going into the bundle sheath cell, or is it converting to malate and is malate going into the bundle?
No.
Sheath? Converting to malate and malate is
going
to the bundle sheath cell. This is false. See, the mesophyll cells of C4 plants do not contain the Rubisco enzyme and initial carbon dioxide fixation occurs there. So mesophyll cell, is Calvin cycle there? No Calvin cycle. So Rubisco is not there. And the initial carbon dioxide fixation occurs there. Is the initial carbon dioxide fixation occurring? Yes. Carbon dioxide plus phosphoenol pyruvate will give you oxaloacetate. So phosphoenol pyruvate combining with carbon dioxide is the first carbon dioxide fixation that occurs in C4 pathway. So what is the second carbon dioxide fixation that occurs in the Kelvin sign. So then this C statement is correct. D, photorespiration is minimized by maintaining a high carbon dioxide concentration within the bundle sheath cells via the C4 pathway. Yes. So all of this process is to ensure that only carbon dioxide is present in the bundle sheath cell. This entire process to ensure that carbon dioxide is present in the bundle sheath cell. Okay. So then this D pathway is correct. Malate releases carbon dioxide. Malate releases carbon dioxide and diffuses back into the mesophyll cells to regenerate phosphoenol pyruvate. That is false. Why? Malate releases carbon dioxide and malate becomes pyruvate. Pyruvate only comes back. Malate doesn't release carbon dioxide and come back into the mesophyll cell. Malate releases the carbon dioxide. Then if once malate is a 4-carbon compound, once a 4-carbon compound releases a carbon dioxide, it becomes a 3-carbon pyruvate. That pyruvate only will come back into the mesophyll cell. So here saying malate releases carbon dioxide and diffuses back into the mesophyll cell to regenerate is wrong. By malate will give out the carbon dioxide and will become pyruvate. This pyruvate only will come back into the mesophyll cells to become phosphoenol pyruvate again. So E statement is incorrect. So correct answer here is C and D, answer number 4. Out. So all 30 questions have been discussed. The ones who have done the paper on. So the ones who have done the paper, can you tell me how much you have gotten? Except for the 2028 batch students, the papers have been marked currently, almost half is done. So you will get your papers handed to you, not handed to you. The PDF version will be sent to you as a drive link, so you can choose your paper. And for the physical students, if you are here, that papers also have been marked, and I will give it to you on Saturday. So on what days will the paper class be held and from what time to what time? So if you are online student, paper writing is held on Monday, 2:30 to 6 p.m. and also on Tuesday, 2:30 to 6 p.m. online paper writing. Okay, if you are physical, then it's 2:30 to 6 p.m. on Monday only. Discussion will be held on Sunday, 2:30 p.m. onwards. $27, very good. 25, not bad. 29, that's wonderful. Okay, that's really good. So for how long will the first term phase go for? 8 weeks. So that would be 2 months, no? So I have kept 8 weeks allocated for everything. Some will have 7, right? Because I starting in September, no? So that the second try students or the 2026 also can join. So therefore I started in September. So most of them will have 8. Some terms will have only 7. So then the first term phase will be kept for 8 weeks. So that's 2 months. Then you'll have the second term phase for another 7 or 8 weeks. And then third term and so on. Final one will for sure be 8. Because I anyway finish the syllabus 2 months before the exam. 20, good. 25. 26. 28. So no one has gotten highest marks. Don't worry. For the highest marks will be given. Okay, it will be provided in a rank. I think totally 46 plus 15. That's 60. 61. 61 students are eligible for ranking. So other students send it to me when they did it on their own time. So they are not eligible for ranking. Out of the 61 students, we'll see. If there are so many who have gotten more than 75, then I will put a top 10 ranking. If not the top 5, I think I'll just go with top 5. Okay, top 5. So then I will put it in the channel like I told you all. Right. So any other questions from the MCQs? Any other questions from the MCQs? So are we discussing part 2 now? Yes, we are. So actually part 2, the problem regarding part 2 is part 2 there is no need to discuss it. It's wrong. I have made a nice scheme. So you can see the scheme here. So I'll give a small break now. What I want you all to do is, here you can see the scheme. Right, so the question is there, answer is here, the number of marks for each is also given. So you can see. So far when I went through the structured essay, I think you all said the structured essay was very easy because all of them are direct questions. Okay. Usually MCQs is what needs to be discussed. But if you have a specific structured essay question that you want me to discuss, please send me a message. I will explain. But everything else you can see I have given the marking scheme and then with the examiner's note all the question 2. Then you have the part B essay. So I will give you a few minutes. Let's say 10 minutes. I'll give you 10 to 15 minutes. Can you go through the part 2 and tell me which questions you want me to do? Right? Right now it's 11:12. We'll start again at 11:22, 25. Right. 25. So it's technically not a break. I want you to go through the structured essay questions and please put it into the chat. That means everyone chat. Huh? Please put to the everyone chat because I can't individually check. Please put it into the everyone chat. The structured essay questions that you want to be discussed. Okay. So if you have chosen all the questions, okay, no problem. We will discuss all the questions. But if you have put specific questions, we will discuss them. Okay, please put it in the chat. So when writing the papers in paper class, should the online students need to join the Zoom session like we usually do when writing papers? Yes, only then will you be eligible for the ranking. So I have given 2 days. Join it, switch on the videos and do the, this for the 2027 paper class students. Switch on the video, join the class, switch on the video and do the exam and then send it as a PDF. And that's when you will be eligible for the ranking. So I have another class at 11:30. So can you, yeah, no problem. You all can go. This meeting is anyway recorded. I will upload it to YouTube and I will share the YouTube link to you. Sir, will we get our papers marked online students? Yes, absolutely. Except for this paper. This was just a practice paper for the 2028 students, which I gave to the 27. But from the actual paper class for the 2027 students, every paper will be marked, okay, by our team. Yeah, I have a team now. So I will mark by our people. Session 2, part A. Okay. Right. So So when will you update the marks to the group? Before this, maybe today? Today might be possible, or today or tomorrow. Today or tomorrow. Okay, then shall we continue with the question? Oh, it's 11:23. Still 2 minutes left. Questions? Question 2, part A4. Question 2. A4. Experiment. Experiment question 1A 4C. Question 2 part C. Okay, shall we continue? So I have questions. The first one. Okay. What is the first step of a simple test carried out to identify non-reducing sugars in a solution? First step. So when we talk about non-reducing sugars, we always talk about who? Sucrose, right? So can you remember the first step? If you put Benedict solution to sucrose, nothing will happen, no? So first step that we do is, we have 2 centimeter cube of the sucrose sample or the non-reducing sugar sample. And then what do? We add 1 centimeter cube of diluted HCl. And see, is that enough? No. Once you add 1 cubic centimeter of diluted HCl, then you boil it. Okay, then you boil it. Boil. Triangle means heating, no? Triangle means heat. Then you boil it. Once you boil it, what happens? This sucrose breaks down. Sucrose breaks down into whom? Sucrose breaks down into glucose plus fructose. And what is glucose and fructose? Both glucose and fructose are reducing sugars. So then they will give a positive reaction with Benedict's test. Okay. So however, after you add HCl, you need to bring it back normal. Then you add sodium hydroxide or sodium hydrogen hydrogen carbonate. Okay. Sodium hydrogen carbonate. So then you can neutralize the solution. But the first step would be to add 1 cubic centimeters of HCl and boil the mixture for 1 minute to get this sucrose to break down into glucose plus fructose. Who asked that question? Is it okay? The one who asked that question, is it clear?
Yeah, sir. Sir, are the values necessary?
Values. Yes, values are necessary. Values are necessary because it is there no, in the tube that I provided, practical tube, the values 2 cubic centimeters given.
Okay, sir.
Right. Yes, 1 mark for that. Oops. So what is the next question? Did you all get this correct? Hydrogen, because it's glycine.
Yes, sir.
Good.
Yes, sir.
This organelle X, organelle Y. Do you remember organelle X is a subcellular component that produces organelle Y? Organelle X is Golgi apparatus, and Golgi apparatus one function is production of lysosomes, which in itself is an organelle. Okay, that is a question I think I created. I can't remember exactly. Can you share this PDF to the group? Yes, I will. I have already uploaded into our 2028 drive folder, but for the others, yeah, I will share it into the group. Then, what's the next question you have asked? Here. How is an enzyme catalyzed reaction affected when the temperature increases above the optimum level? Right. So what happens? What is the relation? What is an enzyme, by the way? What is the organic molecule it is made up of?
Globular protein.
Globular protein. No, it is a protein. When you take a globular protein means now what level of structure are most of the enzymes present in?
Tertiary structure.
Structure. So what happens to tertiary structure proteins when you have high heat?
They will be
Denaturation.
Denatured. What do you think will happen to these proteins at optimum temperature?
It will be at its optimum efficiently, maximum efficient work.
Very good, right? So what happens when you increase it past its optimum temperature? That's when the enzyme starts getting denatured. So if you take a look at the marking scheme over here, you can see when the temperature increases beyond the optimum temperature, the hydrogen bonds, ionic bonds, weak chemical bonds of the enzyme's active sites are disrupted, right? Because they are made up of proteins. The result is in a change of the shape of the active site. Now, if the shape is changed, the entire issue with enzymes or the entire principle in which they work in is shape, complementary. But now if the shape has changed due to denaturation, can they bind to the substrate anymore? Not possible. The result is a change in the shape of the active site of the enzyme, which alters the complementary nature of the active site. The complementary binding of enzyme active sites and substrate molecule is prevented, causing a rapid decline in enzyme activity until it stops completely. So enzyme activity can eventually reach zero when you keep increasing the temperature because the enzyme would have been completely denatured such that they cannot bind to the substrate anymore. So who asked this question? Are you okay with it? So who asked this question? Somebody asked this question. Yes, sir. Okay, right. Sir, is it okay instead of COOH, is it correct if I write R? No, you can't write R. If you're talking about the amino acid, COOH is carboxyl group, NH2 is ammonium, amine group, hydrogen is anyway there, and instead of the R group, you need to write H. Okay, the amino acid glycine, if you remember, the R group is replaced by H. Right, so then, next question was, here. Okay, the entirety of 2C. The entirety of 2C. Okay, no problem. We will discuss it. State two specific sites where substrate phosphorylation occurs during the aerobic cellular respiration of a glucose molecule in a eukaryotic cell. Substrate phosphorylation means what? Production of ATP from breaking down of complex molecule into simple molecules. So where does it take place? Glycolysis, you have ATP production, and citric acid cycle. Didn't we just discuss it in the MCQ? One ATP is produced in citric acid cycle. Yes, so both of which have substrate level phosphorylation. But are they asking you the steps? No, they're asking you the sites. So glycolysis, where does it take place? In the cytosol of the cell. So you need to write cytosol. Okay. For citric acid cycle, where does it take place? In the mitochondrial matrix. Okay. So to get the answer, you need to write cytosol or you need to write cytosol and mitochondrial matrix.
Sir, if you have written the site correctly and the step wrongly, like mitochondrial matrix, and I have written link reaction during pyruvate oxidation.
Ayyo, if you had written just mitochondrial matrix, you would have gotten it correct. But since you have written something additional, you'll have to put it as wrong. Okay, now I have to inform the paper marker to put Nasiha's statement wrong because he might have put mitochondrial matrix correct because you can't, right? If they ask you the site, just write the site. Don't write anything extra. If you write anything extra and if it is wrong, can't give mark because they can't give you a correct mark for a wrong answer. Okay. So if they ask site, stop with site. All right. So now I'll go and inform him not to give the mark. Okay.
Okay, sir.
Cytosol and mitochondrial matrix. You have to write only those two sites. Then state the step related only to aerobic respiration where decarboxylation does not occur. Glycolysis, then you have Krebs cycle, then you have citric acid cycle, then you have oxidative phosphorylation. Okay. Decarboxylation does not occur in oxidative phosphorylation of the electron transport chain. Okay. State the step related only to aerobic respiration where decarboxylation does not occur. However, there is another step where decarboxylation does not occur. Which step is that? What is decarboxylation?
Sir, can you repeat the question?
Yeah.
In glycolysis also decarboxylation does not occur, right?
Sorry.
In glycolysis.
Very good, very good. In glycolysis also decarboxylation does not occur. What is decarboxylation? Removal of a carbon dioxide. But in glycolysis, a 6 carbon is broken down into 2, 3 carbons. So 2, 3 plus 3 is 6. So no carbon dioxides have been removed, right? It just has broken down. But if you go to citric acid cycle, if you go to link reaction, those places carbon dioxide is removed. So pyruvate goes to acetyl-CoA, removal of carbon dioxide. Then in citric acid cycle, citrate becomes oxaloacetate, removal of 2 carbon dioxide. So carbon dioxide is removed. But glycolysis, no carbon dioxide is removed. So over here, why can't I take glycolysis as an answer? Can anyone tell me?
Because it is common to both aerobic and anaerobic respiration.
Wonderful. Because it is common to both aerobic and anaerobic. For you to get here the correct answer only to aerobic respiration. So always read the question and underline the question. So since it's a structured paper, underline it with a pencil. But always read the question twice and answer it correctly. Because if you don't know something and you got it wrong, you can bear, no problem. But if you know something and you got it wrong, then it's a bit difficult to tolerate it. Okay, right. Then, since entire 2C was asked to answer, I will explain it. Okay. State 2 similarities between ethyl alcohol fermentation and lactic acid fermentation. Both are occurring in the cytosol, no? Because both have glycolysis, so both occur in the cytosol. That is true. Both utilize glycolysis as their first step to break down one molecule of glucose into 2 molecules of pyruvate. So if you just put here both have glycolysis as their first step, that is also okay. You will get a mark. Then both produce a net yield of 2 ATP molecules and 2 NADH molecules per glucose molecules. That is also correct. Here the net is obviously important because ATP you form 4. Both must recycle NADH back to NAD plus to maintain the limited pool. That is also correct. So any 2 if you mark, you will get 2 marks. Okay.
Excuse me, sir.
Yes.
If I write 2 ATPs are produced, is that wrong?
Yes, no. Can you tell me why?
Because the net yield is 2, right?
Yeah, net yield is 2. All right. Okay.
Sir,
Yes.
sir,
Excuse me, sir.
can we write like this? Both lactic acid fermentation and ethyl alcohol fermentation are anaerobic.
Yeah, anaerobic. That is okay. But as you can see here, that anaerobic part and the cytosol part has been included to 1 point. So you need to write both anaerobic and cytosol location to get this specific point. Okay.
Sir, I have a doubt.
Yes.
The similarities, can we write like the final electron acceptor of both ethyl alcohol fermentation and lactic acid fermentation is an organic compound, right? So can we write that as similarities?
No, you can't write that as a similarity because that is not mentioned in the text. Because all of these points, if you can take a look, they are something that is actually significant. So saying that both have organic compounds as acceptors is correct. Yes, technically it is correct. But as it is not in the marking scheme, we can't provide marks for it. Okay. All right. So any other questions? So if we write that 2 processes doesn't need oxygen, yes, no problem. But as you can see, the anaerobic part and the cytosol part has come to the same point. So you need to write both occur in the cytosol and anaerobic for you to get 1 point. Writing anaerobic alone will not give you half. You need both to get 1. So this is how strict the structured marking is. Okay, not only the structured, even the essay marking is like that. Okay.
Sir,
Yeah.
out of how many marks is the entire structured essay mark from?
Structured essay, each question is 40 and then you multiply it by 2.5 to give 100. So 2 structured essays you will get 100 and then the 2 essays you will get 300. So in 300 plus 200 will give you 500 and the MCQs is out of 30. So multiply by 10, 300. Total paper is out of 800. Out of the total you divide it by 8 to get your final mark out of 100. So What is the total amount of ATP produced in less active cells? Do you remember? It is only 30 ATP. What about in liver cells and cardiac muscle cells? How many ATP formed in liver cells and cardiac muscle cells?
32 ATP.
32 ATP. What is the reason for the 2 ATP? So that the 2 NADH
Active transfer occurs
The
plasma
two
and
ATPs are used to transport to NADH from the cytosol to mitochondrial matrix.
Yes. 2 ATP molecules produced during glycolysis are consumed to actively transport the 2 NADH molecules from the cytosol into the mitochondrial matrix. You need to write it like that to get 2 marks. Okay. Now, this is the one that most people had issue with. But I don't know why you will have an issue with this because I explained this properly. Anyways, we'll see. A student prepares the following apparatus to calculate the respiratory quotient. Equal masses of germinating mung beans have been added to both respirometers. So this is the respirometer, this is a respirometer, this is a water bath. Water bath is to maintain constant temperature. Right, capillary tube filled with KOH is placed inside A. So then here you have KOH, and capillary tube filled with water is placed inside B. Here you have water. What is the reason for using germinating mung beans for this experiment? That is because they have a high respiratory rate. Germinating seeds are actively respiring metabolic tissues. They consume a measurable volume of oxygen and release a measurable volume of carbon dioxide in a relatively short period of time. Or if we have mentioned that they have a high respiratory rate or rate of respiration, that is also okay, right? High respiration rate. So now, as you remember, when you do this experiment, you leave these two setups aside for 2 hours to see whether there is any movement in this liquid column. So if you use germinating mung beans, you still need to wait 2 hours. So if you use something that is not as fast as mung beans, you might have had to wait much longer than that. Okay. Then, what is the
Excuse me, sir.
Yeah.
Then highly respirable, easy to get observations. Is it correct?
Sorry.
Highly respirable, easy to get observations.
Easy to get observations. Highly respirable, but is respirable a word though? It should be high respiratory rate. Respirable. Respirable is not a word, right? So it has to say high respiratory rate and easy for observation of water column changes. That is also okay, but the respirable, I don't think that you will get a mark for that. It has to be high respiratory rate. Then B. What is the purpose of adding potassium hydroxide to the capillary tube A? What does potassium hydroxide do?
It absorbs
It
carbon
absorbs
dioxide.
carbon dioxide.
So whatever volume change whatever level change that occurs here is due to what? Completely due to oxygen alone, right? It is due to oxygen alone. Then here, to absorb the carbon dioxide evolved during respiration so that any volume or change in the respirometer is solely due to the volume of oxygen consumed. Then, when observing after about 2 hours, there is no change in the liquid levels. In apparatus B, after observing for 2 hours, there is no change in the liquid level. So then what does that tell you? What does that tell you if there is no change?
The respiratory substrate is a carbohydrate.
Respiratory?
Substrate is a carbohydrate.
Yes, respiratory substrate is a carbohydrate. So what does it mean by that? Why do you say it is carbohydrate?
Because the respiratory quotient is 1. So that corresponds to carbohydrates only.
Yes. Respirable is a word, it seems. Okay, means air or gas that is fit for breathing. Respirable and high respiratory state is not the same. Respirable and high respiratory rate is not the same. So we can't give marks for that. Okay, so back to this. So how do we say that it is carbohydrate? That is because these mung beans will produce carbon dioxide and they will use oxygen. If there is no water level change here, that means the amount of oxygen used and the amount of carbon dioxide released are the same. The only respiratory substrate that can do that is whom? Glucose or carbohydrates. Okay. So that is why here for this question, what is the respiratory substrate over here? What is the respiratory substrate that has been subjected to respiration? It is carbohydrates. And mention the reason for the above conclusion. Like we just told, because the volume of oxygen and carbon dioxide is equal. That is because carbohydrate has a respiratory quotient of 1. Okay. Then when observing after 2 hours, the liquid level in arm P of the apparatus has risen. What could have been subjected to respiration? So over here, when you take after some time, after 2 hours, the P has come up and the Q has gone down. So what does that mean? So this respiratory mung beans will take oxygen, will produce carbon dioxide. So can you tell me, does it take in more oxygen or does it release out more carbon dioxide?
Take in more oxygen.
Take in more oxygen and release out less carbon dioxide. So what is the equation for respiratory quotient? Respiratory quotient is VCO2 divided by VO2. So if VO2 is more and VCO2 is less, will you get a respiratory quotient of more than 1, equal to 1 or less than 1?
Less than 1.
Less than 1.
Less than
If you
1.
get a respiratory quotient less than 1, what are the respiratory quotient less than 1 values that you know?
0.7 and 0.8.
0.7 and 0.8. 0.7 belonging to whom?
Lipids.
Lipids.
0.8
Proteins.
belonging to whom?
Proteins.
Protein. So then the respiratory
Proteins.
substrate would have been what?
Proteins or
Lipids or
lipids.
proteins.
It's all lipids. It could be either proteins or lipids. So that is why if you take the last answer, when the observing after 2 hours, the liquid level in arm P of the apparatus has risen, what could have been subjected to respiration? It could be either fats or proteins, lipids or proteins. Right. So that concludes the structured essay discussion. Okay. Any questions? I have discussed everything, right? Yeah, all the questions I have discussed.
Sir,
Yeah.
sir, I received the Unit 3 book just now.
Okay, right. Very good. So others also will still be receiving it. I think there are only like 5 or few people who haven't received it. Others, like 50 others have received it. Okay. So part P essay, that is exactly what is in the textbook. I have given you a marking scheme and I have given the diagrams that are necessary for the marking scheme. Okay. So actually I have added a little bit more diagrams here. Any 36 points, you can get 144 from the text alone. See, from the text alone, you can get 144. For the diagrams, I have given 5 marks. If you have written all 5 diagrams correctly, you get an additional 1 mark, totally 150. Usually, they don't expect you to draw all these diagrams. Okay, I just put it for the sake of completeness, but usually they don't expect you to draw all these diagrams. So if the question is describe the structure of an amino acid, yes, then they might have expected you to draw the amino acid structure. Then describe the levels of protein structure, then yes, they might have expected you to draw this. But again, like I told you, first focus on what? Out of these marks, which one do you think is the most important?
Text.
36 points.
144. Take a look. 144 marks are available from what? The text. So make sure to write the text first. Okay. Then describe the process and significance of mitosis. This diagram you get, I think, 5 marks, is it? How much? I can't remember how much I gave. Yeah, 6 marks. Correctly drawn diagram, 6 marks. Okay. So here you have the scheme. 38, 37 points are there. Any 36 points. Then write short notes on the following. Here there was no diagram, I think. Yeah. There was no diagram. So 39 points available. Any 38. Maximum is 150. So I will share this thing into the group. Okay. Do you have any other questions?
No, sir, clear.
Sir, for the question 1A, 5A. Okay, let's go to 1A, 5A. So the allosteric regulation question, wasn't that challenging?
No, sir.
Not much, sir.
1A, 5A. What should be the answer? 1A, 5A. State a physical difference between saturated and unsaturated. Physical means they're talking about how it looks, a physical state of matter, right? So saturated fats are solid at room temperature, unsaturated are liquid at room temperature. Okay. Chemical difference only, you are talking about the double bonds. Physical is physical state of matter. Then let's just go to the allosteric regulation because I thought that was a nice question. Here, right? This question. So can you see once A is binding to that allosteric site, that is when the active site is properly, stabilized. So then this A has to be an activator or an inhibitor.
Allosteric activator.
Allosteric activator. Okay. Then B is the substrate which the enzyme acts on. C could be the active site or even the polypeptide subunit. Both are acceptable. Okay, both are acceptable, but I have given it as active site. Active site or the subunit, both are acceptable. Yeah, then okay. That's it then. All right. So if you have any other questions relevant to anything else, you all can ask me. If not,
So can C be the enzyme?
Sorry?
Can C be the enzyme?
C be the enzyme. No. If you write enzyme subunit also or allosteric enzyme subunit, acceptable because here the arrow doesn't go all the way to the active site. Okay. Allosteric enzyme subunit, that also I will accept. But just enzyme is not enough.
What about catalytically active form with functioning active sites?
Sorry?
Enzyme which is catalytically active form with a functioning active sites.
That's what you wrote for C.
Yeah.
Why is it so long? Catalytic, again, can you repeat it? It was too long for me.
Enzyme, in the bracket I wrote catalytically active form with the functioning active sites.
No. Within brackets, don't write within brackets. Okay. They will not accept any within bracket answers. No within bracket answers. They will not accept within bracket answers because they know you are not sure of it. Okay. Do not write within brackets. Within brackets is only the marking scheme. Only marking scheme can put within bracket because if you put within bracket with the marking scheme is telling you, okay, they can write this or it's okay to miss it also. But if you write within brackets, they will not accept the answer. I remember some examiner told me that. I will anyways confirm that and let you know, but don't write within brackets. Yeah.
Enzyme subunit having an active site.
One at a time. One at a time. Sorry?
So can we enzyme subunits for C?
Enzyme subunit, yes, that should be accepted. I will let the markers know.
Can I write enzyme subunit for C without allosteric?
Yeah, and for C, enzyme subunit is okay.
Excuse me, sir.
Enzyme subunit having an active site.
That is perfect. That is putting both as the correct. Okay, enzyme subunit. I will let them know now. Enzyme subunit can be accepted.
Excuse me, sir.
Yes.
So I wrote enzyme and within the brackets I wrote active site.
Within the bracket can't accept. I'll go and tell. I didn't include that in my instructions. I'll go and tell now that within brackets should not accept the answers. Okay.
So excuse us since it's the first time.
No excuse, nothing like that. This is not
Why,
A
sir?
level. This is not A level, no. That's a regulatory molecule. I would have given, but no, it is clearly visible that it is an activator, no? Allosteric activator, regulatory molecule only, but it's clearly visible it is an activator. So regulatory, I will not accept. No excuse since it is your first time, because this is not your A level, so it doesn't really matter. But once you properly learn, then only, to learn, you have to make mistakes. Failure is the pillar of success, is some people would have said it without knowing the actual meaning behind what does failure mean, right? Failure is when you learn. Failure is important for you to build proper connections. Without failure, you can't learn. So this failure could come in your exam, or this failure could come when you're learning. So that is the best part to make the failure. That is when you're learning. So when you're learning a content, you try to come up with an explanation or reasoning, and then you check with your teacher or an AI to see, is it this exact reasoning? And then if you make a mistake, the correct reasoning you will remember for a long time. So failure, everybody fails. But the difference between most people and those top 1% is those top 1% fail when they are studying by themselves. The others fail in the exam. Okay, so make sure that you fail in these exams, in these practice papers, fail at your lessons, fail in your studies. It's okay to fail. Fail and correct. When you correct your failure, you will remember it for a longer time. And then in the A-level exam, make sure that you don't make any mistakes. Okay, so no first time, no second time, nothing like that. So if you don't have any other questions, we can conclude the lesson for today.